I'm good. All right, cool. All right. Good morning, good afternoon, good evening, wherever you might be joining us from. We are back on the bottom with the remotely operated vehicle Sebastian, 2.5 kilometers below the surface of the Pacific Ocean at the East Pacific Rise. We're in an area kind of just north of Tica Spire or Tica Vent. And for anyone who tuned in yesterday for our dive, this might look familiar. This is the experiment that was placed down yesterday. So at the beginning of this dive, we just wanted to come back to this experimental site, make sure it's still here, still looking good. It is. The crab has not yet flipped over the box. And we're going to pick up the frame that we left here yesterday, I believe, as well, and then move on from here to explore the area and find an area suitable for experiment number 2. So that's what we'll be doing for the foreseeable future on this dive. But again, welcome. As folks kind of trickle in, I'm sure I'll restate what we're doing a couple times, but we're looking for more of a mobile dive today probably, but also some sampling along the way as we look for a suitable site for experiment number 2. I'll hand it on over to our scientists. You'll hear them kind of on and off throughout the dive, and then if you have any questions, feel free to type those in the chat, either here or either on YouTube or on Facebook, or if you can't quite follow along as we're live, you can still keep tabs on what's going on and submit things on Twitter as well using the hashtag #VentUnderworld. And there's some great posts already there going into a little bit of the science of this expedition. And you can learn more at the schmidtocean.org website as well, where we have an expedition page, including some blog posts that'll be getting put up throughout the expedition from the scientists themselves. So I'll let us kind of settle in on this expedition, or excuse me, on this particular dive.
John?
Right.
Also die Seiten haben beide gut ausgeschaut für mich. Da habe ich geschaut. Das ist okay, weil da ist dahinter das. Ja, das ist schon okay. Am I now? Is this on or off?
It's on.
It's on?
Yeah.
Okay, good. Hello from my side also. I'm Monica Bright. I'm the chief scientist of this expedition, and we just were diving on this site where we deployed the mesh box, the first one, and we're checking out the sealing to the bottom, to the basalt, so that there's no possibility for any animals to enter the box from the side. So whatever has to come must come from the bottom then. The goal of today is that we find another nice location, hopefully very flat, with some small tube worms to select our second site for the second mesh box, so for our second experiment. For this we will first just pick up our frame that gives us the dimensions. With this we can measure exactly how big the place has to be. Yeah, we looked at this yesterday, no? This was the first we looked at, so I think we saw all 4 sites. This is fine. I think we can pick up the frame now. The crab is funny, no? I don't know. Thinking about it. Yeah, the crabs usually jump on any experiments you deploy very fast.
They do
Curious.
like to kind of like investigate.
Curious animals, no?
Yeah.
But they have been doing this on all of our deployments. Yeah. There's another one here, so this is really nice.
The protection.
Well, you gave them that nice climbing wall. You know, it's got the little circles in it, so
Yeah.
they like to climb.
Thank you. Thank you. I mean, the net we're using is 32 microns only.
Yeah, yeah.
Which is basically, of course, letting the bacteria through, but keeps really all animals out.
Yeah, yeah. Oh, yeah.
And
No, I don't think they're
for
like
protection, we made a plastic cover with lots of holes in it also.
Yeah.
And this is what Sabine was referring to, because if the crabs are walking up there and the nets were fine, no, they might tear it a little bit or make the holes in the net larger, no?
Yeah, yeah.
So
Oh, yeah.
So they're not able to grip into that net because it's so small.
yeah, it was as a protection also for deployment, because it's really difficult to handle when you know everything inside, you know,
Yeah.
cannot touch, but
Already crab on there, isn't there?
yeah.
Yeah, no, the crab is.
2 of them actually.
They're already.
Trying to break into the vault.
For some reason, I think they're just very curious.
Either you guys want a coffee or anything, or don't have to have it? Kind of to you.
Yeah. Okay. Okay. We will slowly walk our way to waypoint 5, which is very close, I think, but just during moving over there slowly, we will probably could stop several times just to see if there's some
OBS 5? That's how
OBS 5, yeah. Yeah,
So if
but
you're at 11,
why is it
should
so?
be like here. I think. Yeah, still not getting there.
Yeah.
Just be careful backing up around here with all the
Yeah.
kids.
Is this the ROV disturbing the bottom? Because there's so much, no, because there's so much stuff floating around.
Yeah, much more than yesterday. Eruption.
Yeah,
These are usually the sites that's getting
Yeah, it's
away.
off to the distance as well. I think this is beyond the ROV, but I don't know.
Yeah, yeah.
No, I'm not
No,
kidding.
you're not kidding.
Saw a little bit in your aft. Might still going over it. Just keep rotating like you were. Maybe it's on your left, kind of almost underneath you now.
So you saw that we picked up this frame. You can see it here in our little side camera there. So that frame is roughly the footprint of the boxes that are being used for these experiments, the mesh box. So we'll be using this frame to kind of measure out a suitable area for experiment number 2.
Okay. Let's go back to 11, since it's easy to find. And let's face
Yeah,
the exact direction. And then just go that way from there.
we can just follow basically the cracks where the animals are. This is
Or actually, up right here.
there's something down there, but I think this is steep. Is this steep here? This place here?
Yes.
Yes, it goes just okay. This is also steep.
So get on that, face basically backwards from this, and then let's visually spot a location and fly to it.
Yeah. We just try to go up. So then basically north, almost north, go up here.
So the map is a little bit off right now.
Well, it doesn't matter. I think we just can follow easily the
Okay, so you don't want to go to waypoint. Okay, I understand. That's all we're trying to do is get our position figured out and
Yeah.
head in the direction that you were asking us to
Yeah.
go.
Because there's one here, but this is steep.
Don't come down. Is it under you?
Don't come down.
It's almost
Just-
Yeah, do it down.
Yeah,
You won't- Don't come down.
there's nothing.
I would've,
This we saw.
tried to keep an eye on it. That's the best thing.
Pretty sure. I've got a meter and a half right now, so.
Maybe here. There's something here. It was something further here.
So yeah, let's
I was not.
keep it inside. That way I can know the heading that you
Was it this? Das sind only sehr bullets, I think.
That's way off to your right now.
So again, we're at East Pacific Rise, which is an area that has been
Yeah, 2 pumps
well
probably.
explored.
I don't know.
And so every once in a while you can kind of see these little square blocks. Some of them look a bit rusty or corroded. Those are leftover weights from previous expeditions here with submersibles,
I
ballast
just was
weights.
curious about here because I see 2 pumps here. So this is one here. This is actually a crack here. I don't know, steep or not steep, but this maybe is good. We can check this out in detail. Is this flat?
It's going up.
I think it goes up here and it's more. Is this steep? Is this. Over there you see, I guess.
The area of the East Pacific Rise that we're actually at right now, we're about 2,000 kilometers west of Costa Rica and 1,000 kilometers
Maybe
south
box
of
would be like
Mexico,
this
give or
side
take.
maximum, no? So it's a long, small little thing. Yeah, this also, but I have to look closer, I don't know.
You can go ahead and take the camera.
There are several animals in the area that are going to be pretty common on this stream, on this dive. We of course have the large tube worms that look like
I don't
little
want to
lipsticks
put
or
the camera, I
something.
want to have the camera in my direction.
But we also have smaller tube worms, all those little white
This is like
squiggly
your
bits on the
direction,
rocks.
Marty.
Those are also little different tube worms there. We have the squat lobsters,
but
which are
we
those
switched it. For
kind
me,
of
we
round
switched it and it's not
crustaceans
switched.
that you'll occasionally see one sort of swim in front of the camera using its tail. We have one actually that came up and said hey to our situation camera. Sometimes they'll hop up there. Looks like he's drifting down now. Right there.
I cannot.
Coming down into the view of the science camera there as well. So every once in a while, we'll see those land elsewhere on the sub. They really kind of just go wherever they please. Another animal that we'll see a lot of are
Right here where the other one
the
is.
eel pouts. So those are the kind
we
of
see it.
longer
Okay.
eel-like fish that we see. And those are probably the most common things. We'll also get these mussels, like you can see there on the screen, bivalves.
Can somebody maybe help me with this? Turning this around
Oh,
in
yeah.
the opposite direction again, because I cannot make.
We need to toggle a dip switch on it, yeah. Let me, let me see it there. We're just adjusting the camera controls here. Anyone who might play games and you understand the conflict that might arise between people who want the inverted controls and people who don't. Just a question of comfort. And so we're just swapping some controls around for people. So for these experimental sites, what we're looking for generally are some areas with cracks where there's venting fluid coming up from beneath the ground, similar to where we placed experiment number 1. And so this is, these are hydrothermal flow coming up, so warmer waters. Now these waters do come in other areas. So they're pulled in these little intake areas, and then they're expelled as heated fluids elsewhere, which is what we're looking for. We're looking for where they're venting up out of the ground. And the idea is that we place these boxes, this mesh box, over top of the venting water area. And we're actually looking for organisms that may have been brought under the ground by the intakes of the flow and then have survived the whole process of getting heated up and ejected back out at a venting site. And so that little mesh box over there, we're hoping to leave that there, I think, for about 10 or 11 days. And then afterwards, we're going to see sort of if any critters have come up into the box, which is otherwise sealed down to the seafloor as best we could. We used some caulking and blocked up the cracks and everything. And we're going to see if perhaps the venting itself is delivering fauna to these vent sites. So as we look around today, we're looking for another suitable site, an area that is flat, an area that has venting. We'll probably take some temperature readings, but also it's got to be an area that we can get a good seal down. So level and flat for sure.
This is, I think, not, is this flat? Is it? This is like this. This is like this, no? Steep, no?
It's
What do you?
hard to say. It looks close to flat.
Really?
Can you put it down?
I can. It's. Do you care about the angle of the square right now?
No.
No.
Also, I mean, the crack would be good if it's in the middle, but
That rock right there is not
yeah,
sliding or anything.
this is true, yeah. Actually, this is true.
Yeah, no,
Let's just see.
that's pretty good.
If you move away the rock, would you see just a little bit?
Just set it aside.
Yeah. Okay, thank you. Let's just
I
check
don't
out.
find it too bad, to be honest. Because there's like here a large one, but the only like basically there's only one crack running through. So you'd only need to see like here, and there, and here. But not now.
I just go in the main.
Here it's good.
What a
There
bit
might be a
sorry.
small crack on the
And
top
the
right.
top right here, okay?
Yeah.
This one here. Here's a small one. But very small. And here we have like the super big one.
Yeah, you
So
would
it's
have
one
to.
you would have to.
I mean, if you would shift it tiny little bit, but you're gluing basically the animal on it. You have to glue here now, right on the animal. So this would be basically the spot where you glue. Can you maybe switch on the laser so we kind of have a feeling how deep this is, because this is big and deep. You know,
But
this because the animal is in it.
yeah, I think it's like about 4 centimeters, 3 to 4 centimeters, I would guess. And then it's, of course it's going down.
Yeah,
See the lasers?
I see. Thank you.
For anyone just tuning in, you are watching live ROV footage, or remotely operated vehicle, from about 2.5 kilometers below the surface of the ocean at the East Pacific Rise. We're currently looking for a suitable site for an experimental deployment. If you happen to tune in yesterday, for our dive yesterday, you saw one of those experiments being deployed. We began this dive by revisiting that site, just making sure that that box was still in place. And now we are looking for a suitable area for experiment number 2.
Yeah, this must be correct, but this is small and it goes wider.
No question there. No, there will be easy. But I think the position of the finger is good.
Would you like another waypoint here?
I'm thinking. I trust this correct too. If you follow, I just try to follow a little bit. Just follow the other side a little bit. But I think here it stops also.
So I think the spot for the frame is good, but I'm kind of a bit worried if this is a good. I think we can seal it, but it doesn't seem so super active here because like they reach down a lot, you know.
You don't know what is below now because the crack is like if the crack is about 4 centimeters, the animal sticks in here. Also the other smaller ones. We have to break this open in the end now and pick everything we can, so it could be good, but it's very different than the other situation, so maybe it's good to make something a little bit different. But we would have to go with the pry bar in here on the side, no, afterwards, after the experiment is done here, and really rip it apart, sort of. And this here you cannot enter so good, but start from this side, you know, and try to collect everything. Let's take a waypoint. I think we look more, but
Like I said, probably to the right one, that's probably the rock is a bit looser, so to the right one, if you zoom out, to the right. You mean this here, or?
these are two.
Yeah.
Then you have 3 large cracks. And also this is as a shoulder. You cannot put it here. You could put it here, but this is. Let me see.
What would you like to name this location?
We call it waypoint. Let me see. The next one is 12, I think.
There's 12, I think.
12. We make a waypoint here and just potential site or potential experimental site. Because if you take this here, well, I mean, I cannot see this. I don't like. I like this one less than this one, but yeah, just take the frame and we can put it there again. We find it. We look more. Look more around.
Can we zoom out a bit that we get
Yeah, I do.
an overview?
Yeah.
Where it is?
Sorry,
With or without the frame?
with the frame and then this is all.
I can't reach it. Okay. Well, so. Then we put it right there.
This is not far away from the other side. Okay, I leave it. So we find it. No.
I think it's okay.
No. I'm sure you don't measure much temperature here because it's much more below,
That's a
but
problem.
sometimes you see something you don't even need to measure really. But it is a potential place.
Yeah.
This one, this is all kind of steep here. I don't know. Yeah, this is, we can go here along. I mean, if you circle around here, we can look at this sort of. Doesn't not necessarily need to be that close,
No.
but this is more active because also of this, you can see it in the surrounding. This is can you go here? Let us look here, everywhere. I look here. This is really like plates, no? It's almost perfect for.
Look around.
Mm-hmm. If you can. This is big flow here. See? I don't know. Here.
We frequently talk about shimmering water, and that's what you're seeing here. So that's evidence of venting in this area.
We could do. Let me see if I can look here first. Like this is relatively young and fast growing because it's so thin, the tube. So can we try to place the frame and see if it's steep or not? Because
Yeah, I'd like to just bring up one point. I don't know if you see in the camera there's the previous box. So depending on which way we have to come in for some of this work, we could be very close to disturbing
Okay.
your previous.
then look, I think maybe we go on the top and look along here and maybe on the other side, like here, and go along here. See, like this, we can go along here and go up here. And it could be even that if you turn around and, face the other direction, really the opposite, not south but north. You can see this. Yeah, this is really close.
So you just follow along that.
Yeah.
Okay.
Also,
Yep.
this is the top really, no? So we can look on the other side and the top and go along here, so because this is really close to the other side. This you could see shimmering better than I like better. It was not the first one.
Yeah, we're right over the other side.
Nothing empty. Right over the box or like that's
One also. What was this? Wie war das reingehauen? Oder die schaut so rot aus, da ist sicher irgendwas. The thing of marks we could just wedge against here. Von Marks sein. Und können wir dann einstecken. Okay. That looks Carl, how do you see the evenness of flat, also steep or not steep? I don't know.
Yeah, it looks a little steep to me.
Okay, then we go.
A little bit of that area over here.
Hmm.
Right. I don't know how that works for you guys as far as like there's a lot of stuff going on there. Right?
Which area did you mean? Uh-huh. Okay.
Maybe right here would be flat. This looks sloped. This looks sloped.
This,
There's like a little spot right
this
there.
here.
Might be okay.
Yeah.
You mean that one?
Yeah.
Okay.
It's still hard to tell.
Yeah.
I'm just guessing.
I know what you mean. Yeah, just look at this first. Hmm. This is just iron. I think. I mean, Eisen. Also roasting. Oh.
Got a couple questions here. We've got obviously what might the temperature of the venting water be? We'll probably be taking a temperature reading at some point, I would think.
Yeah.
So then we will find out what the temperature here is.
This is so
And we've got folks coming in that are curious. We keep talking about this experiment, and you might not have seen yesterday's dive. So yesterday we came down and we placed experiment number 1, which took the form of a mesh box that was placed over the seafloor over an area of venting water, hot venting water coming up from the seafloor. And this mesh box was actually sealed to the seafloor over that venting area. We used, we had syringes of caulking to get in there and seal up the cracks and everything. Quite a complex operation, but we revisited the site this morning and it looks like it was a successful deployment, which is pretty cool. But it's not the only one we're doing. So now we're actually looking for another suitable site for experiment number 2. And so what we're looking for is venting water, and what you see on the screen right now is kind of an indication of that venting water, that shimmering water. We're also looking for a flat area, because the box has to just kind of stay there. It needs to not slide around. An area that's not too rugged or torn up or too craggly, because the box needs to seal to the seafloor. So these are all kind of considerations that we have.
We could try.
And the scientist on the mic right now that you're hearing on and off over there, that's Monica Bright.
see which ones.
And this frame is another tool that we're using in our search for experiment site number 2. This frame's basically the footprint of the box that they're hoping to place. So we can use this to kind of gauge whether the box would actually fit in an area, how it might fit in the area, and what cracks might cross into the footprint of the box that we'll have to consider when they're sealing it down to the seafloor.
Analysis where das nach unten floppt. Also doch, ja, this is not possible and also it's very
No, I don't know.
Yeah,
Can you zoom?
this is, yeah, I can zoom, but it
So like here it's okay, here it's okay. And here would have to fill up, like basically, but like volume.
no, you don't see it because it sits on the muscle.
Can
You
you
have,
move it a bit?
yeah,
Like one like bit that's not sitting on the muscle. Just give it a bit of
So to kind of pick up of what's the experiment doing, right? Like I mentioned that we're sealing it down. So after the mesh box was in place, then we put another box on top of it, and we filled the entire experiment with a dye that would stain anything that might be living inside that box still, but also just kind of a general stain of the area. And then we're leaving that box in place, the mesh box, excuse me, we're leaving that in place for hopefully 11 days. And when we come back and recover, we are looking for any organisms that aren't stained. What we're curious is, do the venting fluids themselves transport animals to these areas through the ground?
we know that we can crack it, no? Also, let me see. Oh, sorry, I'm doing it wrong. I'm doing it. Okay, let's just look. So deployment would be possible. Site is relatively flat. It's very different from the first one. This is also not bad because we wanted to go for a little bit of diversity. Let's measure the temperature. Or basically,
How stable do you think is the frame?
yeah, how good can you work in this area like you're now for all the stuff we had to do yesterday?
Okay, so yeah, I think everything should be fine. I don't see any issues.
Do you think it can slide easily? Because it looks a bit. Can you like try to poke it so you see it's like sliding?
Is it sliding in this area or not? Or when it's sliding down? Or
Yeah, the frame itself, it doesn't seem like it. There's a tiny bit of slope, but
But not.
it doesn't seem like it's very much.
you can see from the other side there's also a little bit.
I think that rock is good.
Actually, I think it's good.
This is the surface thing. This is not. There's no crack. It's just the surface thing where it comes off.
Yeah, I mean, it says.
No, that's good.
Sure.
Yeah.
I think.
Yeah, that's good.
And the bottom of your mesh box
It's also
gets a little bit more grippy and even, so I think you can even do better.
Let's just take a waypoint, a frame grab. I'd make it a little. Yeah, let's take a frame grab.
just as drift.
Yeah, yeah.
What's that point? Just drifting.
12 we have already. Let me see. Did you have to write the
I was about to take the finger out and I didn't move my mouth.
waypoint 12 we have, so you're waypoint 13 now. And just write also good, more flow.
Quotations of an experiment.
Right.
I mean, once we're sitting, then there's no DVL lock on the bottom.
I make a close up also.
Hard time on this.
So when you lock this in,
Like
then
Vegas,
I
you can sit back and place it.
then now take a highlight frame grab, and then I zoom in for another highlight frame grab.
And quotation size.
Well, DVL, I guess you have to wait for you to
Yeah, let's pop up and just creep our nose.
Yeah.
So basically you're looking straight out.
Can I move the camera?
And see if it gets better.
Okay. Let's look like this.
I'm moving this up.
Okay, good.
No, no big deal. I'm just letting you know, just so you're not wondering why the camera's moving.
Mm-hmm.
See if we can get a better location. Navigation.
You know, the other one on top, what you just saw, this is about the thickest, the other one, the first one we had, and I'm really worried about this. Because if, I don't know how,
Yes.
let's say, we don't know how much we can break or not, so it's just a guess now. But I would try to sample and the control next to it and then leave it as it is. And then we will see how this will break. This will be the water, the rock, and the sample. We don't need to take the next or the next, but we need to take one of those, you know. Right?
Okay. It's not working.
But we can move from the measurement of this one, if we really say, we take this, we move to one of them, so then the control sample.
I think we just put down 12.
Yeah. Because we would leave the frame. We would leave the frame. It's like that. So, it is not far away, but it is still very different from it.
Fine enough.
Far enough, but also a little bit also different. The frame, yeah, we will do that. We would leave the frame. We made already a waypoint, no? We take pictures and we take temperature, and we would leave the frame here, yeah. If this is. And one possibility, if this is not too, this is probably steeper, this no, because this is smaller. This here. This would be smaller but steeper, I think. Then this one is not so small.
I thought she said
We're leaving the frame, but we're taking the temperature.
But this here.
Probably don't want it too far.
All right, so as mentioned, we're gonna take some temperature readings here. So for that, the pilots need to use one of ROV Sebastian's manipulators. And actually, we have like a little sheath that the heat probe, the temp probe is in. And then they'll just kind of be able to hold that down in the flow that we see, and we'll be able to read the temperature off of it. So let's figure out how warm this water gets right here.
Let's see. No, I would not want to take it away, but potentially we could hold some of the water sample in it, or we take a little bit bigger one and take a water sample in it. Let's see. It's moving up. I'm not touching it, but the temperature moves up. You can go. What do you start?
The temperature measure.
Okay. Hmm. Ten. Mm. It's not gonna go higher. Maybe we can try here. Okay. Yeah, this is basically the hot place where they sit right here. So it's 10 degrees, it's not... Yeah.
Wir außen da hinten waren, was wir gerade am Anfang gehabt haben. Ich glaube, der Dreier.
Ja, Obst 3 ist das, was wir gesammelt haben. Also wir können nur zum 5 schauen, aber dann sollten, sonst müssen wir das wiederholen.
Ich zoom out.
On here?
Mhm. On the waypoint.
Share here. Because here we have only looked like in the first video, but we never went
This is
back.
not, this is the rubber. Yeah, but this is the, yes, this is the rubber. This is the number 3 is the rubber, and it was looking like this. And the 5 is actually very similar to the number 11.
We selected waypoint 4 yesterday, or the day before yesterday.
Yeah, this is this, and, but this 4, 5, and 11. We are today at 11, no? And 4 is very close to it, and 5 is also good, and 3.
Yeah, this is what
This
I
is,
want to check.
yeah, but this is the rubber below. This is not with the plates. Five is, when this is all close, 3 is here. We haven't looked at the 5 again.
Yes, no, the 5 has been, we are at 5 basically. Almost. So we're very close together.
So our scientists are currently just taking a look at some of the waypoints and locations that we've put down in this area. It sounds like we saw about a max of 10 degrees C here, 10.4 it looks like. But it sounded like that might be a little lower than they were hoping, so it's possible that we'll be scooting to another location.
Good for rough, large works and must maybe place we can flatten smaller part better. I don't know. Yeah, let's go over there. Slowly, maybe we find something
Maybe 3
better.
and 7?
Hmm?
Over to 3 and 7? Or,
3.
okay.
3.
Sorry.
3 is this. 3 is this. Is also super close.
Just to check out. Okay.
What?
Because we have been here, like we haven't looked like only the first day. It's good just to have like now the different view that we have. To
Yeah.
Yeah.
Yeah, you need something small, and
So we're gonna,
we pick up the frame, we find it again, I think.
so you wanna,
We,
okay, we're gonna take
we
the frame
beat-
with us? So we're
yeah,
bringing it
yeah, we take it with us, yeah.
back.
And I think there are more areas where there's just small. This we can do, I think, but
Yeah, I just would like to see that what we've seen in the first week because I don't remember well enough. So,
Excuse me.
let's see.
yeah, I think we just have time for this to look around
Yeah.
more. No? I would rather take this second one now than, so basically this one than the first one.
Yes, me too.
But I think we were excited about the number 3, but this was compared to this one, so now it's too large, way too large. And it was not, I mean, maybe I'm completely wrong, but I think it was this rubber thing. So, yeah. Searching a little bit more. This we could take, but we can always come back now. Look at more.
So we're gonna have to move the boat,
Mm-hmm.
might move with us further, I'm gonna wait for it.
Wie sitzen die Oceanschutz und nicht oben? Die sieht man nicht drin. Schau. We are sitting right. If it goes down in both directions.
Yeah.
I did not realize this.
Yeah, no, no.
It's like on the kind of on the top.
It's because that's why it's flat.
Yeah.
Bridge RV, I got a updated waypoint.
And this is this really lush area where you don't want to collect them. This is huge. This is very huge.
Bridge
Yeah.
RV.
This is
Bridge RV.
there's lots of stuff here. Uh-huh. This is
do it kind of real slow because I can't go in the red circle.
Yeah.
So,
We enjoy the view of thousands and thousands of tube worms and mussels in a relatively large area, actually, I think. And in the middle there's a yeah, this is what is new. This is all stocked barnacles. They came in kind of late after the last eruption.
And when was that eruption? I think you had a date on it recently.
2005/6.
Wow, okay. So since 2005/6, that's where we're seeing all these little stocked barnacles coming in.
Everything came in.
Wow.
It was flat. So basically, this was almost the center of the eruption
Yeah.
where we are right now in this area, and it has been mapped really carefully how far, basically, so the extension of the eruption is well known,
Wow.
and so we know that, of course, everything you see in here is less than 18 years old.
Yeah. Yeah. Yeah.
Also, you know.
Yeah.
And that does not mean that everything settled immediately one day after the eruption. So
Wow.
you can see by the masses of really large tube worms and masses of mussels, this is a very active site.
Yeah. Yeah. Yeah.
And then we know that about 40 species occur in tube worm aggregations, 20 of them small, 20 of them large. And mussels is even more, no? Mussel aggregations have more species.
Yeah.
So it's not many species compared to other environments, no? If you think about coral reefs or something like this, or seagrass, but they occur all in really, really high numbers.
Yeah.
Stop turning right now.
that's where we have a mark.
Wow.
Not there.
Probably underneath it, so you're kind of on top of that side. Usually you want to be a little bit away, so always in front of it. Just be careful. There's some bottom.
I see.
There's nothing you can. This is also 10 meters higher than our last
What was the depth?
mesh box.
Oh, got it.
No, this is, you cannot do anything here. I don't know, we can. Is it possible that the navigation is a little bit off?
Yes. Yeah.
Okay,
I think
that means we are not at
So
our
here's
old
what I
place.
suggested, we're not flying backwards.
Yeah.
Try to avoid that as much as possible. Let's rotate around this,
Yeah.
take your turn,
Yeah.
we'll have a better heading, and fly this all forward. We'll be able to just see what's coming at us
Okay.
instead of trying to.
So go around this fire like.
That's what I would have done. I would have went to the right, taken my turn out, then I'd have been here, and then every everything is in your line of sight then.
And then go the other way.
Yeah. And I guess you don't really have to go all the way around, there's just turn if you'd like, but let's do everything in front of us.
See, there's also something here below. I think that the place we with this ops free waypoint, I don't know, is this high or low? No, this is also high.
Let's just follow that little path and see if we see something that could have been what we flagged.
Currently, we're going over a lot of these mussels. You also got the squat lobster. Folks were asking what the little crabs were. Yeah, so we've got a couple different crustaceans here. I have seen a shrimp or two, and then little swarms of amphipods that kind of seem to hang out in the flowing water itself. And then these squat lobster that are everywhere. I'm actually looking right now on our other cameras. Sometimes we have little hitchhikers that jump on board the sub for a bit.
This is a thanks.
Is this this is the sponges? Yeah. So these are some sponges that were placed here. Think kind of like a shower loofah sponge or something, providing a texture and a substrate for little organisms in the water to kind of settle out on there and colonize those a bit, just as a sort of a collection instrument there. And in fact, one of those chains of sponges is underneath the mesh box, again, to kind of provide a substrate for anything that comes into that box through the flow to sort of settle out on.
Yeah. I mean, we must have one.
Let's see where that one was relative to where we're
Okay.
trying to go.
Yeah. You can waypoint it again. So we are gone. Otherwise, we could just again, and then we can look. This was which dive? This deployment was the first one, guys. Yeah, that we. Op 6.
Oops. No. That's
What?
no,
Yeah.
6.
No, sorry. Collection trooper muscle come close to Op 6 deployment sponges on basalt. Wait a second. We don't have one. No, what?
Can you find these waypoints on the
The sponge was from a previous dive. Yeah, these were from our first dive here, I believe. And we were speaking about them earlier. So those little amphipods, there's kind of this cloud that's much denser immediately over top. But you can see lots of these little bits of, they look like dust, these little motes. Some of them are swimming on their own. They're changing direction. They're going against the current. So those are those little amphipods that are just cruising around. It's crazy to me, the size of these swarms. They're just fascinating to me, sort of like a murmuration that you might see in something like starlings or another little bird.
Op 7, it's the sponges in the worms.
That's good to know, so then,
We just need to head to the northwest. Okay. The map.
Yeah, they just want to, we just want to compare then the Op 7 to the long lot we're doing now, because this is a little bit off.
Now, while the sponges were put by us on a previous dive, this little block, little cube that you see there, that's the ballast weight from a previous expedition of some sort. Some sort of submersible would have been down here with those. Could be Alvin weights, could be something else. But those basically get left behind, and as you're going to see, they start to deteriorate.
is that an octopus?
It is indeed an octopus. Good spot.
Did you check now the Ops 7 versus?
We've got a little octopus just barely
We did.
over on the side.
So now we have an idea of where 3 should be. He's heading there now.
Okay.
Real briefly, he was over there. You might have to go back. He is not in our path of travel.
Should be.
This is all this one. There was nothing that goes on from. Das ist, was ich geschrieben habe, die Scheibe immer nur wenig, aber es ist eigentlich alles da.
We were at 3, so it was 7, like follow the line.
Das sind die Oberhand, die 7 Quarter Quiver, Small Rock, Mist, 7.
More amphipods there in a little swarm.
Hammer, Browns 1 to 6, there's nix. Dann haben wir 5, which you have really good flow. 4 best. Die waren einfach die ein größerer, ein bisschen kleiner, aber auch der kleinere war viel größer als das, was wir machen können. We could also look here at the bottom a little bit. Because maybe this is good. This is again one thing, but maybe we can go somewhere here. Is this up or down? Or? This is up, no? This is up, but this is more flat, no? This is what we. This is actually, I think even this one is over here. This is a bigger one. This is a smaller one. But we did not look at the steepness at all on the first dive. We did not. I think this one.
Octopus again.
We could just look in between a little bit what it shows, because now we know a little bit more what we are really looking for. But I mean, this and this is also a very big area.
Very
I don't
tiny.
know I don't know if the octopus is that small. No, we have to go. No, this is all. This is.
Now we know that our first observations were. Yeah. Now we know more.
Now we know more, and we have to go back in this 5.
So anyone who might have been joining us for our last expedition, the Octopus Odyssey, this is very different from the moose octopus that we were seeing brooding in those areas. Hiding a bit from us there, behind that rock.
This is what we waypointed the left here. This was waypoint 3, ops 3. If you, I don't know.
Yeah, so we're obviously seeing the offset, so we can just keep that in mind. If we put waypoints on an offset map, I think with other waypoints that are not offset, I think everything's just going to be super confusing.
Okay, we just know now we took 1 waypoint for, or 2 we took already, but this is potential places for deployment
Okay.
today.
Is this a new place then, or are you thinking this is 3?
This is 3 for sure.
Okay, yeah. but
So,
I
okay,
don't
just,
think we should take another one.
okay, I just wanted to know if you want to take it. We don't need it. Okay. We circle back slowly to the 5 area because
I think 7 might have been on a different dive, so they could have an offset on that.
to the today 13 area, yeah.
Okay, so come back to 13. Get up.
I think we can extend a little bit in directions and then.
The frame now there,
Frame is there.
nothing is moving.
Ja, well, 13.
Lass mal hinlegen, wenn wir den nehmen, den Spot.
Genau, ja. Machst du
Man
gleich.
kann auch nicht finden. Naja, wenn wir dazu kommen, wenn wir hinten in die Richtung, müssen wir wieder schauen. Ne? Das ist jetzt erst 10, ne? Also, wir haben schon noch genug Zeit, weil wir müssen nicht nördlich gehen, wie beim ersten draufkommen. Ne?
Richtig drauf, ja. Wir haben jetzt noch keine gute Stelle.
Ja, ich bin mir nicht so sicher. Wenn man muss, kann man jetzt so nicht nehmen. Ne? Also,
Genau.
aber
So if you're just joining us, or maybe you joined us a little while ago and you're not quite sure what we're doing, this is actually live footage from about 2.5 kilometers below the surface of the ocean. It's coming to you live from a remotely operated vehicle named Sebastian, and that's being driven remotely by folks up on the research vessel Falcor 2, up on the surface above it. We are currently at the East Pacific Rise, near an event known as Tica vent. We're about 2,000 kilometers to the west of Costa Rica and 1,000 kilometers south of Mexico, give or take in there. This is our, or this dive, excuse me, this dive we're looking for where we could place our second experiment. So yesterday we placed experiment number 1, which you can actually see up there at the top of this ridge. I think that's experiment 1 there. It was a little mesh box that got put down, kind of sealed to the bottom. We're leaving that in place for a while to see what comes up underneath it, because that is placed on top of venting water. So obviously, as we're looking around, we are seeing areas of hydrothermal venting where hot water is coming up from below the crust here, or below the ground, the surface of the seafloor, and that hot water brings minerals with it, obviously. But something we're curious about is since there are intakes into those systems where water is sucked in to the below the ground, and then there's areas where the water comes up out of the ground. We're interested if any of the fauna in the area, perhaps its larval stages or its gametes, are being transported through that flow of water. So under that box, we're going to collect that in, like I said, about 10, 11 days time or so, and see if anything might have been transported up through the flow into the box. But we do have 2 more of those boxes to place during this expedition. So for this dive today, we're scoping around. We're looking for a prime location for experiment number 2 for the second box. And the sort of thing we're looking for a flat area. We're looking for an area with venting. It needs to be pretty level so that the box doesn't slide around. And it can't have too many cracks. It needs a crack or 2 because we need that venting. But if it has too many cracks, we won't be able to get a good seal down with the box, because the box actually gets sealed as best we can to the sea floor here. So if you're just joining us, that's where you join us now as we're exploring around an area north of Tica vent, looking for a location to deploy experiment number 2 on a future dive.
Are we? Okay, that is good. Pretty close. This, let's move a little bit, like just to extend a little bit in circles around our mesh box, sort of a little bit, and look if anything could be possible or is there. This is very small, this place.
Ja, ik vind niet dat we nooit verdwijnen.
What? I don't know. Because here you see, I mean, there's.
But if we continue now north, as we're doing right now, then it's all the basalt and we don't see anything. If we continue like more to the, so basically all our interesting stuff is like in this area here is like this kind of.
Yeah, that's why I say like make a circle.
Places. And here we get the rubbery stuff with.
Yeah, I know.
And here it's like basically the with the huge.
Yeah, that's Tica prime,
Yeah.
or sort of. But I mean, like, if you circle,
We have done this in the first dive.
yeah, but it doesn't matter. I mean, I still would like to see how much, because here's nothing, no?
Yeah, we've done it
And then we turn, basically we turn because this is empty, and we go, here's something, for example. This is something.
Which we've seen today.
No, because we have not been here today. We circle now, the box is on the left side. So here, if we look here for something, let's just see. Sometimes there's something, but not quite good. Let's go here on this place.
That's to the left on your pen and tool.
Mm-hmm. Yeah, I know where it is. This is steep, no?
That's pretty steep.
This is steep. This is... The box is over there, I saw it already. This looks good, but actually I looked at this already and then
Yes.
we decided that this is too steep. So, but this is the crack that goes like this. And then we can go... Maybe this is flat. Or is this also steep? Because this is the ridge, no? We cannot do, on this ridge we cannot do anything. But here, because we needed just a small place here, worms. Haben wir den angeschaut schon mal heute? This is the place. Are we back at the place or just does it look deep similar? Where are we? Didn't we just put this on here? Our frame? Is this not the first one we did?
Waypoint 13.
Waypoint 12. Could it be? Or am I?
The one we just did today, I think, is waypoint 13. Right?
We did 12 and 13 today.
I have no idea. That's a good thing we're here.
But this is not 12.
What?
I'm looking to the waypoint 12. So this is the variable different.
No, that's different. Yeah.
This is waypoint 12. I have the long lens here.
I would bet that this is where we put on the frame the first time.
Waypoint 13.
No, not 13, but the first was 12.
The first was 12.
Yeah, this one that I showed you. So, yeah.
This was 12.
This is 12.
No,
No?
you must have look at here the picture. At what Andre is showing.
This is 12.
It's different. It's very similar, but the masters are not there.
Yeah.
So we're currently looking for a location to place the second box. I believe there are 3 boxes total for this expedition. So we'll do this one, place it, and then repeat this process for the third box.
I mean, should we make them here waypoint? What's in the back then?
Actually, maybe let me look at this one first. Yeah, this one, let me look a little bit and then we'd make one waypoint, or let's look a little bit together. I mean, this I don't find so good because the worms are really stuck deep and the crack is not defined, so we have problems to figure out what do we seal or not.
So in the Tica press area, I think it's the only area that's flat enough to do this. And we've been a bit further north, like on the first time, but there it was even, things were even larger. But we have not looked, for example, up at this wall.
East wall,
There's
I
more
think,
stuff like further north. But I think that in this area,
we have actually in this area.
we have seen it, I think. And we know basically it's like our waypoint 12 this today, the 13 was I think the best one up to now.
Yeah.
This could be another one, this would be waypoint 14 if we do a waypoint.
Let's
And I kind of have the feeling that we've seen this area, that we know where we could place things and it's not getting better than this.
yeah, right.
So what we could do is also like decide on, for tomorrow, like let's do for example waypoint 13 where we also measure temperature.
Yeah, I know
Collect
what.
samples, but then I would try to move further north to just explore a bit more, because I think this is what is here we have seen. And for tomorrow we have a place, I think.
We need a place today.
Yeah. Like, but for the experiment tomorrow,
No, today.
but for the staining and stuff.
Yeah, but it's starting we
Yeah,
need to do
today,
today.
yes,
So this,
yeah.
that means we need to choose one today,
Okay, yes.
and we can look at. I think I would like either this or this. Or we can try this to measure here.
I'm more worried about the third deployment. This is what I'm thinking right now.
Okay, yeah, but
Find
the
a
third we
place.
can still think, yeah.
Yeah.
Okay, I'm thinking that we saw it here. This would be another spot. We put the frame there, then we take a waypoint, then we measure the temperature, and then we take one of the three and start to work. So, if you can,
Okay, so deploy the frame.
yeah, right. You're coming now like this, but maybe it's the other side is better to sit. I don't know. Also, it's not so big. Okay.
Are you interested in this right clump or the one to the left?
Yeah. Just put it on the right one. And then if we measure on the other side, I will go through this crack to measure the temperature and we might move it.
See that
Not so good, no?
backside, there's that rock.
Yeah.
Not good, yeah. Maybe the other one is better,
Maybe
the left one. Mm-hmm. Yeah.
that's the left part.
And for this we would have to use a lot of here. If you shift, maybe you can shift it a little bit so that evens it out, because here's a hole, not, but here I think this would be probably all covered really by the silicone that we have. Let me just see, and then we take. Could you turn a little bit like,
Turn the frame.
Mm-hmm.
Which way? Counterclockwise.
yeah, a little bit like this? Maybe that evens out a little bit more, or not, I don't know. Yeah, just. No, it's not. It was better first. This is too high. You could go. Can you go down again where you were basically? Let's see. Now this. It's not even at all. It's
Well, that, there's a gap
Yeah.
under this center of this frame.
yeah, I
right
just see
here,
this. I go.
But everything else is real close. Yeah, exactly, right there's a gap.
Because the frame is there, you cannot see good. We just, let's take another waypoint and let's take measurements, because in worst case we could probably chalk it, but
Okay, waypoint 14.
It's 14.
yeah.
We want you to fly up or just let it be where
Did we measure the temperature at 13?
Yes.
Can you look what it was? 12 was, I think, 10 degrees, and 13, I don't know what it was.
Yes. It's 10 degrees.
And the 12 also? Also, I mean, the
We
previous.
just measure temperature once.
We did not measure temperature there. You forgot?
No, I measure temperature.
Okay.
Okay, the waypoint is taken.
Okay. We try to take temperature.
As soon as he lands out.
Mm-hmm. Ja, es ist irgendwo, muss man glaube ich sehen, wenn wir sehen, wie unter dem Handle vom Frame ist es so durchspalt. Ja. Ich kann jetzt nicht, wer es jetzt 7 jetzt mal, dann können wir wieder. Und dann muss ich aber trotzdem die Temperatur, das ist vielleicht das, was am schlechtesten geht, aber nur zu sicher. Ja, das sind die alle, die sind wieder auf, bei denen nehmen wir jetzt los und dann dann immer zweiten. Aber ich schaue jetzt gerade mal wegen der Temperatur von. Weil eigentlich kann man die Temperatur messen vom zweiten, weil irgendwie.
Ja, 20 Grad.
Das war das 20. Okay, was nicht schon. We don't have to look at it.
Also rather 10
The
degrees.
10 was
The
the
10
first
was-
one, and the 20 was the second one.
Like you talk about today?
Oh,
Yeah,
today we
today.
just measured once.
One, yeah, the
Okay,
first we
then-
didn't measure,
Yeah.
but also
This-
because it was so deep in, we couldn't see any shimmer. So the first one was like, the first today was not so good.
And the second was the 20.
second was
Was
the
the
10
10.
degrees.
Okay, good.
Yes.
Okay, got it. Because I did write it down like this, but I was thinking maybe I forgot to write it down. So
No, but you know, at the waypoint 12 that we took, you could see that the tubes like really only stick out and they reach down very deep. It was the one with the very big crack.
Yeah.
and we couldn't see shimmering on top. That's why we said we don't need to measure temperature, because if we don't see any shimmering,
Okay.
it's
The 13, and here we measure a little bit, and then I think we go back to the 13, and do, and
Yes.
basically do the next collections or stuff.
Yeah.
One of the next ones, but
Yeah.
not super close. And that we destroy.
Yeah, the temperature probe.
This is the temperature probe now that comes, and I'm done.
How active is the area like further north, like after Eastbourne?
Yeah. I don't have my. If I sit down, I have to get coffee. I just get down this picture. I just like simple, but I do it back and look for the. I bring them other maps.
Have you set the waypoint already?
No. The waypoint did we set? Yes.
This is 14.
Yeah, we did.
Yeah, yes. Are you seeing any fluids coming out of it?
Oh, absolutely.
So again, we're just taking the temperature here, see what the temperature of any water flowing out might be. And you can see it, it's a little warmer than the ambient around it.
The best of was 3.5 on the left side.
Pick up the frame and we go to waypoint 13.
right, so we're gonna pick up and move back to one of the other areas that we've already looked at on this dive, but I think we're just gonna check that out a bit more in depth. Again, we've only got a few of these boxes, and they are tricky little setups, so just finding the ideal spaces for these boxes is definitely a goal of well, the expedition as a whole. But for this dive in particular, we are looking for the location to put the second box, or experiment number 2. While we're scooting, I will say that our connection's looking pretty decent today. But let's check out what the weather looks like. For anyone who tuned in yesterday, this is certainly looking a little better out there. We've got some blue sky going. Lots of clouds though, so I'll be keeping an eye on those, because again, those can certainly interfere with our stream. But you know, it's not looking bad out there today. And hopefully the experience shoreside for everyone joining us is looking about the same.
Yeah, it seems like everyone was lost. Oh, no,
Yeah, we adjusted this one.
but it was near kind of the edge of the ridge, so you just went over it. So maybe if you want to get your safe heading and then face that and just ladder roll or like fly along the ridge until we see what it looks like.
Oh. So on 13 we can see the box.
I think so, yes. So maybe somewhere in there.
I think it was here.
But while we're on the way, does any of this look decent on this side here? Kind of steep, isn't it? Yeah, we should try it. Try it over there. Or is it here? Actually, I think it's right here, right? Not the 2 pumps? Oh no, maybe not. No, those
Can you
are 2
open up?
jackets.
Can you open up waypoint 13?
Should have gone like right.
Experiment.
I think this is the area.
Yeah. And we put, here it is. This here we placed it on. This what it is. Here we had it. Yes, I think this is the place.
Yeah, there's that muscle. Seems right, because then there's offload rock.
Yeah, here.
Yes, yeah, that's it. It's good. Good enough. Yeah, that's it. Yeah, it's that one.
Yep.
So this is the area that we will tomorrow put the mesh box on, so we can place the frame now here, I think.
All right, so it sounds like this might be our candidate for tomorrow, placing experiment number 2. We're just going to put that frame there, which marks the footprint of where the experiment will be.
But we need to get a good position that we see again like where all the cracks are, like we did on the. We can move it a bit towards us, the frame, because now it's like also here on the crack. And if we can avoid that, so if we can punch it a bit like towards this direction. Like that. Sorry? I think that's good, yeah?
Below the 2 points there's a gap, no? On the right side. Here.
Sorry?
Here. There is like a big gap, no? Or not?
No, yes, there you would like.
What if I slide it a little closer to us, the same angle, but just.
But that's, yeah. Since we took those with it. Yeah. We can frame him. Yeah.
Zo, je zou hem met gedacht. Oké.
That maybe?
Did you try this already, like in this, like this?
Yes, we tried
Okay,
that,
that
and
was
then
not good? Okay.
no, it was good, but then we have like also this crack here.
But this is not
This
so
is not so
because
good
we
with the
need to see
tubes.
the-
Yeah.
Okay.
That's okay.
I brought some chocolate.
This is where points 13 or 12. 13. 13.
Can you shift this a little bit, like so this is straight? And then we move it just down a little bit if possible, or is this not possible? It was not possible?
No, we had in the beginning, but in the beginning then there was the crack up, and I basically told to move it different.
Yeah, okay, but when we play around a little bit with the best, no? So basically between here and make this line maybe straight, because we cannot try to shift it as down as possible, so we don't reach this upper crack. Yeah, this is good, I think. Yeah, looks good, like this. Yeah. A little bit notched up here, so a little bit. The muscle.
Basically to glue the muscle.
Okay, okay, stop. Yeah, good, super. So we have actually this crack here and here. I see nothing here, I see nothing up there. Good? Good. Highlight frame grab. You did already like this with the frame in it like this because we will leave it? No? This is basically, I don't know, this you have to seal here. Can you go up a little bit? But this here mainly, and this here, because there's nothing here, but we can look again a little bit closer, okay? There's nothing here. I go. There's nothing here. There's this crack here. Can you print this picture you just took? Is this possible?
Print?
Yeah. Here. There's nothing here. There's nothing here. Let's go here. And this is where we measure the temperature, no? Yeah, if we want, we can seal here because, but I think this is not a crack. But if we want, we could put here, but I think 2 spots, like this one. Oh, sorry, now I'm confusing myself. Like this one, and the other one.
Can you see the cracks in this one? Can you see the cracks?
Yeah.
Yeah, we also need like, so from this also, also from above, that we see it like this, like it's now.
Yeah.
Just make like,
I let you.
that we have a bit of an overview also that
Yeah.
we see the boxes also in the back, so that we, you know, so that we have
Yeah,
this orientation.
that we have to go from this side. And
Keep the handle on.
yeah. I think this is much easier than the other one. You just have tuk tuk, basically. So
Can you have enough pictures? Can you zoom out with your camera a little bit?
I can here. Okay, got out.
No.
It's
Can
okay.
you take another
Mm-hmm.
another frame?
So the temperature measurement we took before. Now is the decision if we go right in there to take the water sample or if we choose the one we will collect. Because here we get less, here we'll get more, and this is the same. Just see what we could grab.
So you want to collect the one next to it?
That maybe not really next to it, or we could try, no? We could try, oh sorry, now I'm confused again, myself. Yeah, I mean, this one would be good to collect because it's smaller. This one we could try also one of the others because here we could put in the pry bar here, you know? It's very close to where we want to be, really. Or we could try to start from here just and try to get something. But if we take this one, we would go in here with the water sample, no? I think this we can try. So that means we have all the pictures, we have the measurement, we have the frame.
You do not want to try to take the tube bones from the frame.
No, no. Because you completely disturb it, no. We will break everything. I don't want to.
You're not removing them?
No.
Fish box?
No, because you cannot get them out without removing the place totally. That's what we tried the first time. I'm sure. I think I want to sample here the water and take as much as we can from here.
Move one more to the left.
Let's see, we can also move one more to the left or to the right. Let's see which one is. It's basically probably what we have to do then anyway, you know, because we have to work our side in, but we can also go in this one because this is following the crack also. This here. This is.
Your point with the
If you don't take this, we can take this, or let's see if you like another one better. Or it's pretty similar, no?
Just gonna move that on.
Yeah. But that's we did the first time also. There were just less there. It's you destroy also the crack and everything, you know. So I like this one better than left. What do you like, Sabine? This better than we start from the other side of this to first we do this.
Same as disturbing.
So I still would take, just for comparison, try to take a temperature measurement. So this, we take, I go like this. Okay, so we see it. Take a highlight frame and call it experimental site control. So everything we do, we are going to call experimental site 2 control. Instead of the 1 and then the known also. Okay, this is the highlight frame we have, no?
Yeah.
Grab. So we take, the next thing is we take some, we probe in here, temperature.
3 seconds. One move over back.
Suction sampling, almost blocks.
Okay, let me just turn the.
Ich schaue das dann noch mit meiner
I'm just gonna
Liste.
move over a little bit.
Ja, ja,
so we get
ja.
closer.
genommen. Anybody wanting chocolate? More for me? Anybody here chocolate? Oh, chocolate. I grabbed the whole thing, I found it, and I said I need it for
All right, so let's see what we got.
Brauchst du wieder was müssen wir aufspritzen?
No, das was ich finde.
All right, so it looks like we've settled on that for the site for experiment 2, which will get placed tomorrow. I believe that's the plan. You can see experiment 1 there in the background, top of frame. But for now, we're going to move on to some collections here, samples and measurements. It sounds like they want to leave the worms and mussel intact inside the experiment site there for experiment 2. We did remove them for experiment 1, but it also kind of tore up the ground a little bit. So for experiment 2, it sounded like they might want to leave that more intact. But regardless, we're going to be taking some samples and measurements here for the next little bit.
Det er kanskje ikke nedad.
Ja.
Snap that things out.
Okay. Okay. Let's work a little bit. So, the others not me.
You want more?
Okay.
All right, while we get in place here to take our measurements, just a quick recap for anyone who might just be joining us now. We are back down on bottom, about 2.5 kilometers below the surface at the East Pacific Rise. And we are in the process of getting some samples and collections next to an area that we just selected for experiment number 2 to be placed down tomorrow. So yesterday we placed experiment 1, which we might see here when the camera turns a little bit.
Yippee.
But the experiment takes the form of a mesh box that was placed over some hydrothermal venting and sealed down to the substrate here, sealed to the rock. And the idea is we're going to leave that box in place now for 10, 11 days, somewhere around there. And when we come back, we will hopefully see things inside the box that were not there when we placed it. Organisms possibly that are being brought up, that's the idea at least, being brought up in the hydrothermal vent, in the flow that's coming out from below the ground. But we just selected a site for another placement of a box like that for experiment number 2. And now nearby we're taking some measurements and sampling.
You're not even very far in, so we know that this is.
If you went down, that went deeper. 7.3.
But if you are joining us, if you're watching, and you have any questions about what you're seeing or the work being done here, feel free to type those in the chat if you're watching on YouTube or Facebook. And you can also follow along even when you can't watch us live. You can follow the hashtag #VentUnderworld, #VentUnderworld on Twitter. And that hashtag has got some great little posts so far looking at some of the science, but also many of the people that like watching our streams will also post up screen grabs, and we'll post little clips as well of the things that we see and what we discover.
No, this is great, thank you. We're just welcome.
I'm gonna put in whoever.
Yeah, that's a good one. That won't fit in that. Leo, don't go
Right here.
Left. Temperature 11, okay. Good. So the next thing. Yeah.
come out a little bit.
This we don't need.
No, we're putting the probe in there
Okay.
just because it's
Yeah,
easier.
it's good.
The little holes are cracked.
Let me see.
The holes in the crack is the fact because it's just really hard.
Okay. Good.
It's gonna fall.
Okay.
See your arm with it?
I can see it.
Almost. There you go. No. Alright.
Good.
Okay.
We will do this water sampling in the blue bag.
Okay.
Right. Let me see. This is right.
Right
Right here where basically the measurement is,
Okay.
and after this we will try to recover the animals
Okay.
with your pry bar and
So we put the probe in with the left hand then?
Yeah,
so this will be
Experimental side too.
controlled. This now is all controlled. Experiment to control.
Okay.
So the temperature is experiment to control. Yeah, but we don't have it. Yeah, if you can put it in. Okay. Waypoint 14. Yeah. So this is what is observational.
It's a comment and will be attached to the temperature.
All right, and if you've tuned in for some of the earlier dives to this expedition, then you have seen this instrument. This is for some water collection, water sampling.
That's sampling now.
Sampling now with the
What will be experimental side two?
experimental side to control.
So on the back of remotely operated vehicle Sebastian right now, we actually have a little bag. You can see like right there, below one of our thrusters. And so this bag's currently empty, but ultimately it's going to hold about 55 liters or so of water, or up to 55 liters, I suppose. So that's what is being deployed right now with our manipulator. Is the end of that system. And so we'll kind of put that where we want to draw water from and turn on our little pump and slowly fill that bag with water. This is being used sort of as a control for experimental site 1, or excuse me, experimental site 2, which we just kind of decided upon.
Yeah.
Yeah, almost clear. If you got cold or something,
I got where we were.
Wait a second. In here, you have to write.
You want it above it or inside in this?
We have no specification. Just here we just have feedback, no?
yes.
Okay, then you write down 15 muscles and 2 drums. Exactly where I choose exactly the one where the temperature was like this, no? Okay. Good. Okay, so where do we go? 1, 2,
How
3.
far down do you want it?
Yeah, I mean, if you go in here, like where you went, basically it's sort of where you went with the temperature.
Okay, so
It doesn't need to.
Alright.
and then
Hmm.
give you the set. Zoom in.
Okay. We've locked it in, now it takes a fingers. Yeah, when we're in. Okay, let's see. Where are we?
Bring it deeper into the side. There we go. There we
Every
go.
time I freeze pasta,
So you can't see the next inch.
I want to dig in there. That the highest temperature was actually above. It wasn't above down.
Yeah, but we are sampling basically the microbial community. I don't know if we clog up, I don't know what to do, but a little bit deeper I would like to go if possible. Yeah, this is deeper. Mm-hmm.
Alright, how's that?
Okay. Highlight frame grab and switch on the pump.
All right, so we've got the tip of the tube in there then for where we want to collect the water, but now of course we'll have to turn the system on, which, as you can imagine, right, it's its own hurdle here. A lot of ingenuity and different approaches to science in the deep sea, figuring out how things will be deployed, how things will be recovered, and even just how things will get turned on or off.
Okay.
To experiment or to bag them?
To bag, yeah.
So we need to that lead back all the way. Turn on first.
Turn on first. Turn on first.
SO50625.
So I'll give you
Okay.
not too bad.
Okay.
More. Is it my phone? Thank you. I am. I just needed to see it better.
More, 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 more. More, 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 more. More. Oop, good.
Please. Out of box text.
So,
You go to the other one, not to the other side.
Out to the back and then maybe start.
alright, so
Every time you do it's the same. Because now it's not this and that.
So, well, experiment first,
Yes.
then back.
Yes.
Okay.
Yes. External.
Okay.
Okay.
Just to make sure it's cycled. Yeah, understood.
yeah.
More.
Hold on, I gotta move this another way.
Also. Yeah.
That's good.
More. Also now that you put it back then, no?
Yes.
Okay.
More.
Go back.
Yeah.
Please. And maybe they start.
Monica, I've got to watch.
What should I watch?
Watch to start.
Yeah, yeah. I have it.
Not yet.
Yeah, yeah.
Okay, now to start.
Right.
This is how. Now I must move this back. Sorry.
There's a question about the brown things here, what they are. They are bivalves, but these are mussels here.
From the hose. Also then from this. So we have this outlet. Outlet of, I don't know what this is called, handle, outlet of mesh bag, of blue bag, sorry.
So now that the bag is turned on, we will actually see that slowly filling. It's going to take a while though. So I will probably put that just in the corner.
Now we can see if something stick.
No, we cannot see because I wanted to have it better in we have to look here.
Yes. I'm trying to zoom out here to see what you've talked about. Let's try and get out of it. So 5 minutes will be 11.
In 5 minutes we have to kind of wait. Wow, this is better. I don't see much, but it's still a good view. This you can take a picture also, because it shows better where we took it. I think I don't see any movement.
But there is difficult. There is
Yeah, I know. Actually, there is some movement.
And so what will this water sample be used for primarily? Because it's a lot of water. It's like 55 liters.
Yeah, so this will be the water sample between two worms and mussels, and we measured 10 degrees Celsius. We will collect about 55 liters of this. The first 4 or 5 liters go to the virus group, because they make an experiment to study the influence of viruses on the microbial community, which is also in the same water, and they need to be really fast and take the first samples. Then the second sample will be the sample for sequencing actually all life that is in there. So we will have probes, primers for bacteria, archaea, fungi, small eukaryotes, and also animals in case they are in there. And you don't need really a very big sample for this. The biggest part is about 40 liters that will go to the animal group, that will have a large amount for sieving first, and then also fixing it so they can sequence the individual animals if they want. And the last sample goes also to another, what is the community functional doing, so the bacteria and the archaea that you suck up, basically what the functional do. Which means then in the end, basically we have a really good picture, what is the water between such clumps. They are just basically growing on this vent fluid that comes from the subsurface, and that will complement samples in the end we have from just the surrounding seawater that is cold, and also from sampling the subsurface fluid directly, which we will treat exactly in the same way as the others, and then we can have a kind of a good picture which community, so which species basically do they share, and which species do we only find in one place, you know. And I think it is not pumping, and we will flip a little bit around.
Okay. Okay.
No?
Yes.
Yes. I will stop this.
All right. So they're gonna fiddle with the switches here on the pump and the bag. Try and make sure everything's going there.
The pistol would a little bit up and then back.
Yeah. You know, what we could do also, to move the outlet a little bit up so we can observe it, and when we see that the pump is really running, we move it a little bit further down.
Okay. out of there, then screw it over in place and then shut the nozzle. And,
It's not clogging it, but we can observe it better. I mean,
be sure that
no, I'm sure you can suck, also you can get the water in. It's just easier when we see
Okay,
it. Yeah, just as good, and then we move it back.
just from Baltics,
Yes.
they're wagon. Okay. You want to kill a light there? That's not good.
And if you were joining us for yesterday's dive, when we were deploying the dye to stain inside the experiment, this system was also used for that. We had a different bag on with the stain inside it, and then we just flipped the pump in reverse, and that's what we were using. And then when we were done, we actually sucked that stain back out of the box, so it wasn't just filled with the dye.
Down just a little bit. Pull it that way.
Okay.
Turn it on as well. We'll look first.
So we're just watching now to see if we can see any water flow in.
I think so, because you can see the tentacles are getting pulled in.
I think it's working, but we have to wait a little bit longer till we see it here.
Yeah, he's good. Yeah.
I think it's working, but we cannot see it. Yeah, you know why? Because the others are super quiet, and this one, the chrome, so this animal is reacting to the flow a little bit. See, the one below does nothing, and the one above and to the left is also no movement whatsoever. So, yeah. We wait a little bit. Yeah. When we are sure that I think it's yeah, we just have to take the time, otherwise we switch it off and try again, but I have I don't know. Is this bigger now? I think it.
So the bag inflates quite slowly, so we're all kind of just keeping an eye on it, see if we
I think this is
notice it inflating at all.
Yeah, that part looks like
It was noted that this worm directly in front of it seems to be behaving differently than the other worms around.
I think this one is the only one. See, well now this one is coming, but this was the only one now moving.
yeah. Minutes.
This is a male and this is a female. This one Anders would easily go somewhere.
Yeah.
Because they're so close together. Is this not bigger now? You don't know?
Let me change picture, no change.
But it's only
7 minutes.
I can switch it off and we try one more time.
Yes.
We just can try it again.
So that was.
I switch it off and we try it again.
Same picture. No, this is 5 minutes ago.
Can you make this big?
You look back.
Yeah, but it looks the same. Yeah,
It looks the same.
All right, so comparing the image of the bag that we have now to the image that was taken about 5 minutes ago, it doesn't look like it's pumping. So we're gonna try the old turn it off, turn it back on.
I don't know, do something like air conditioner, do some like strings, close to the, and then if it's successful, or even like a, another thing is called camera, you got 2 ports, you can see it. Okay, you see we've got some idea, put it in one of those, so you can see it. Yeah, yeah, yeah. That's like a, these are applications. Yeah, let's do it. Also like that. Alright. Okay, now the order again out it.
For the.
No, for the one. The bag and the experiment.
Yeah, it's not.
No, the off one.
Alright.
Off on.
Yeah, and then again, back, back, and if not work, the water. I don't know, then it's switcher, something probably.
No, this is what? No, it's not with the animals. This is not the problem. No, for sure not.
I'm stuck.
It's, yeah, but it's the valves or whatever. It has nothing to do with the where you hold it.
Alright.
Yeah.
Okay, now out.
Do you have job where you see the frame good?
As I printed.
Ah,
I should be there.
okay.
I can
And you can also.
I don't think it's gonna.
Huh?
It's just in the road. It's not occasionally we'll just
Play.
let's see if it's.
Oh.
Yeah, it's not gonna be. That's what. Just a little.
No, it's bigger? Or is it just...
The destination or the start of
What? Let me see.
Okay, good.
Back to the experiment and then back to the
Yes, yes.
All right, so as a reminder, this is the bag that we're trying to fill at the back here.
It's okay. Leave it. Can you your picture of two spots?
Sure what?
Of
Alright, so go back to the,
I don't know, we will see what it takes in back.
alright, go back to the back.
Yes,
Gotcha.
yes, but now if you have a picture of these spots.
Can you have it there? Is it on the set? Can you use it? No, he's using the set. We'll wait until once soon as he switches, we'll move straight out. Yeah. Yes. Yeah.
While we're trying to turn this back on, welcome to anyone who might have just joined us. Looks like we've got folks from all over, which is pretty cool. If you are just joining us, you're not really familiar with what we're doing here. We are currently streaming live footage from the remotely operated vehicle Sebastian, which is currently diving about 2.5 kilometers below the surface of the ocean. We're about 1,000 kilometers, excuse me, south of Mexico and about 2,000 kilometers west of Costa Rica on the East Pacific Rise, near the Tica vent area. And earlier in this dive, we kind of cruised around. We were looking for an area to deploy an experiment. We actually deployed one of those experiments yesterday, but we do have 2 more to deploy, so we were looking for other sites today. We have since found a site for tomorrow's deployment of experiment number 2, and currently we're just kind of doing some samples and measurements in the area right next to experimental site 2. And this bag at the back of ROV Sebastian is what we're trying to fill. And we're doing that with this probe here, which leads all the way back. In fact, I can show you kind of a different angle here. So there you can see the manipulator holding the probe down into that clump of tube worms there, and then there's a little hose that comes off the back of that. That flows back to the bag at the back of the ROV. And then also in this camera angle, you can actually see experiment number 1, which was placed yesterday. And that is in the form of a mesh bag, or excuse me, not bag, a mesh box. That box is actually kind of sealed down to the rocks. We had to fill some cracks there with like caulking and get the box sealed down real well. And then we used a dye to stain everything inside that box at the time with the dye. And that way, when we come back in about 11 days or so to recover that box, we'll be able to hopefully find some organisms in there that may have arrived after the box was put down. which scientists are interested to learn if perhaps the venting seawater that comes up from below the ground in these areas, the hydrothermal venting fluids, if those actually bring with them organisms, larvae, or even the little bacteria that thrive in these environments. And it looks like the bag might be pumping. Yeah, excellent. All right, I'll toss up the different view here. So you can see the bag's a little more inflated than it was. And on that probe shot, we can also make out every once in a while some of those little particles
now it's working, and now we can go a little bit further down. Yeah.
Yeah, some of those particles are making their way into the end of the probe. So now that we've got it on, with that good old flip it off, flip it back on, we are going to tuck the probe back down into this clump of tube worms and continue sampling about 55 liters of water, which earlier there was an explanation by Dr. Monica Bright that water actually gets used by many different teams for many different aspects that they're examining at these vents.
Do you have any floating gears? Yeah. Would it fit on a Cornish ID hose?
Yeah, I've got it from last time we did that.
Perfect.
I got two of them.
Sitting under your desk.
So if you take a picture of this, then I give you
That's what
also,
I'll use, and we'll put that
you
in
can
line.
write down,
And then you can see it
we make an
actually
observation
operating.
afterwards, just like above, sucking above muscle, also between community or something like this.
Lovely, lovely.
And then we
Had a question about whether or not experiment 1 and 2 were basically the same setup. And experiments 1, 2, and 3 will all take the form of this mesh box deployment, with then dye being applied to everything in the box at the time of deployment. So yeah, they're all roughly the same setup, but we have to find a different site for each one, and there are factors that we really need at those sites. We need venting fluid, first of all, but in order to get the box sealed down, it needs to be pretty level and pretty flat, and if there's too many cracks, any cracks that extend under the footprint of the box, we're actually going to have to fill those with this caulking that we used out of syringes. So if there's too many cracks, right, for us to fill, that's not a good site either. So yeah, the 3 experiments roughly the same, but being placed at different sites. So different, a little bit different considerations here or there, but all 3 are working towards the same general idea.
Oh yeah. So we use one of these, then we can see the actual flow.
Yeah,
So we're going to try and add that in.
that we put this something in between,
Yeah.
and then we can go on this like
Exactly. Yeah.
like
Whenever
between
we do
the,
it in the back, we could actually see it. Would that be
like here.
yeah? Because if we put it in front, there's a chance we get broken.
Yes, yes, it's a good idea.
Or we have it further up, so it's in the
Yeah, but
behind
then
behind
yeah.
the pipe is secure.
here or here, but if it this, you should see, no? I mean, the camera is not so good, but
No, but at least you would see something.
I'm sure you would
It's
see
not. It's
something.
not a super fast pump, is it?
No,
No.
it's made on purpose, not
Okay. Well, that's what we're doing then.
super. We just have to tell Tiki where he will mount. This is super. Okay. They're arranging now this ship to shore. They were talking with Felix because Paula will be in there also in this ship to shore with the Children University.
Okay.
So, and then I think one newspaper from Austria, they want to report a little bit on this, and they were talking already with Felix, but they sent me questions also somewhere. So good. Now the crabs are coming. Yeah, they're always curious when something happens.
Oh yeah.
Don't know really why.
Nosy.
Nosy and thinking about
food.
thinking about could be food, yes, no, we don't know. But yeah. This with this.
That's a good show.
I'm just gonna get okay. What I will print out also is the community that we're gonna sample, not this one, not this, we sample this one over here.
Okay.
Do you have a picture of this one here?
Many pictures.
Right, we have many, and
Right, now I need to check.
you can also give it to me where you see all.
We saw this one, that's
No.
very nice.
No, but I want to see the community we sample, the whole, so basically more overview of the area or something.
Just
Oopsie,
drop this.
it's good. Very good, very good.
What's the problem?
No, it's not. It was just.
It's like sperm. It's all from
Did
sperm.
you do something, or was?
Zoomed out.
It's in there.
But did you see this flap?
Yeah, no,
So
film,
then,
yeah.
yeah.
You know what it was?
Probably fertilization could be.
Yeah. So there's two possibilities. What that is. The one is sperm bundles of the giant tubworm. They release just sperms in bundles, so they're
Right.
sticking together, and then they find their way to the females, which are close, no? Or it is the fertilized eggs that are released that make basically then develop in the water column to the larvae, and then finally, when they are big enough, they need to find a good place to settle again, no? But it's observed quite often because it's reproducing all year round and do nothing else except eat what the symbionts give them and produce
Reproduce.
offspring
Yeah.
as much as they can. Yeah. Right. Okay. Yeah, it's with the highlights, if you can select for the highlight videos, for the highlight frame grabs, then if that is not disturbing anything, because we still have 20 minutes, we could fire all 3 Niskin bottles and then this is done.
Yeah, sick like this.
We will make sampling.
But she told she doesn't need. It's just
No, she wrote what she wrote she needs. Oh, but wait a second, I have to confirm. Maybe I.
That was the discussion that we had.
So Clarissa no skins, right? And then she said yes.
While we're doing this. Just to say.
Yeah.
I wrote Clarissa no Niskins, right? And she says yes. Okay, good. Then we don't need Niskins.
No Niskins?
No, I wrote. I read the message wrong.
You want to confirm?
No, it's okay. No, she said yesterday also. I just wanted to make sure. No Niskins, okay?
No skins.
No Niskins, good. No Niskins. We still have 20 minutes then.
So this one, Monica.
Hmm?
This one is good for your purposes.
Yeah, right. This is good
Yeah.
because then I can mark. This is the control side. I get the
Okay, I will
right
print.
back because I need a marker.
Yeah.
Oh, can't it,
All right, yeah. That's fine.
what is it? Okay. You already seen this markers?
Huh?
You already seen this markers?
I saw, I just glanced at mine. You want to close your look at them?
No, just wondering if you guys.
I think that when we did finish the experiment yesterday, I went over that direction and they were like, okay, don't go over there because there's already stuff going on. Don't want to disturb it or whatever. So went over there, saw that they were there,
right, to anyone who's just tuning in now, you're watching a live stream from ROV Sebastian, deployed from the research vessel Falcor 2 of the Schmidt Ocean Institute. We are in the East Pacific Rise off the coast of Mexico, but we departed from Costa Rica. But Mexico would be our closest point of reference. And we just have a water sampler that you can see in the shot here. There's a close-up in your top left there that you can see. And we're filling a bag, which is located on the ROV. Just switch to another shot here for you guys to get a view of that. You can set that on this SD quad. There we go. So there are about, I believe, Monica, we have about 15 minutes left, would you say, of filling the bag?
15 minutes, yeah, 14 actually, exactly.
14. No, I was going to guess 14
Okay.
too. Should have gone with my gut. But yeah, once we finish collecting the water samples here, we'll be moving on to some other areas. So feel free to take in all the sites that we have here. There's a lot to look at in this small area, but we'll be doing a little bit more soon enough. Nice and solid, yeah. we'll check in on the bag here, see how much has left to go until it's full. Have a close-up of where we are collecting the water from.
Shutting those doors for elevators going down there? Caffeine free.
Keeps it a lot quieter. It's much better for all. I'm just gonna do some.
I went through on these that way. The next thing off, let me so you can see.
We got Turkish meat pie, Turkish vegetable pie, that's pretty good. Got salted caramel cheesecake. It's not bad.
Turkey meat pie or Turkish?
Turkish. It's like a beef thing. It's all right. You're into that kind of thing. So we're bag filling early.
Yippee.
All right. Find a good spot now.
Yeah, this experiment spot is here for tomorrow.
That's gonna be number 2.
This is number 2, and this is our control community. We will sample after we sample the water. This is the challenge of the getting the rocks, getting into the cracks.
I saw your post that the staining thing worked.
What did I write? I did not saw that, does it?
No.
I don't see what you wrote.
The staining thing worked.
No, yeah, we did it. Yeah.
How you doing, Chris?
I'm not bad, yourself.
Come here, dude. So we should just be able to cut that twice. Whoa. Just fall over there, dude. Cut your hose and just stick that right in there, huh?
There you go, sir.
Oh my God, that's amazing. What's the 4-hour look like?
Isn't that really bad? Not bad enough.
Beautiful. I kinda feel like you're good on the server. I think we've seen West Wall every time.
Yeah, maybe. What I thought this
It's
morning.
not mine, it's hers, but if she
What I didn't see this morning.
she will find it, I lost it in my cup. The muscles look so immobile, but they are funny. One time a colleague made an experiment where they took a clump and they brought it to bare basalt They were waiting, and by the ending of the cruise, they were back.
Really?
Yep.
Why don't they like, oh, because they need the vent fluid.
Yeah, it's actually, they don't need it even really, so just a little bit above the ambient, just a little vent fluid they need, and they found it. So they were moving on back. You think this is so, they're not sessile really.
What's this on their shell?
This is all limpets.
Limpets?
Yeah.
You know what's interesting here is all the vents we were at the Atlantic were covered by the shrimp. They had this
Yeah.
sensor on the top.
Yeah, I know.
Those aren't in the Pacific. Or
Yeah,
what?
no, not here. Yeah.
But
They always continuously go in and out of the vent flow, no really hot, out, in, out, so they're basically cold, hot, cold, hot. No.
I guess I understand that.
But
pretty
at the
much.
vent and in the Atlantic, there are no two worms. I mean, there's, so it's
It's crazy how diverse it is.
yeah. This,
All over the planet.
yeah.
They don't have these little, what are those called, feather worms?
This here,
Or is that barnacle?
this is,
No, not the riftia, the smaller.
these are cephalids, they are related to them. These bristle worms, they're all bristle worms, but they have nothing, they are on sort of periphery. Tiny little, flow maybe. And this is actually different species of tubon. The one you see here with the, this is Oasis, Vera humana,
What are their tubes made out of?
chitin.
Like, so they're kind of flexible, like
Yeah, they are, again,
squishy.
yeah.
Yeah.
Yeah. And those two species and another species we sometimes is also here, but I haven't seen it yet. All the three species share the same Symbiont species. So Endorhiftia persephone is the Symbiont species, also the bacterial species name, and they live basically as free living bacteria with other bacteria everywhere, So, and when a tubworm larva settles, there's a really small window, and then basically the symbiont enters the host, and no other bacteria get in, only the symbiont, first of all, which is already cool. And yeah, and then they just basically, and actually the way they enter is also very strange because they directly infect the skin. So they go basically through the skin, try to find the place where they make this organ later, or find that place, and then this organ builds. And when the animal grows, the digestive system reduces, so mouth closes, digestive system reduces, and the symbionts take over the nutrition. So it's really,
That must have taken a while to evolve, huh?
yeah, right? This is true, I think also. But
So
when
these are eating, these are filter feeders, or is the
those
vent
things
fluid-
here,
Yeah, the riftia?
if their filters only basically picks up oxygen, nitrate, hydrogen sulfide, CO2 goes in through diffusion, everything the bacteria need for chemosynthesis, for producing
Okay.
food.
So it's
They
not,
transport with the blood vascular system.
it's not eating, amphipods or anything like that. It's only
No. So
chemosynthetic only.
yeah, only, basically, absolutely exclusively.
It's got very red.
This is because,
huh?
yeah, because this,
Is that for the oxygen transport?
they have, this is also another cool story really, because they have this blood vascular system, like transporting the oxygen everywhere, but the hemoglobin they have, so like our hemoglobin has one docking site for the oxygen, no? And if we breathe in hydrogen sulfide, the gas of stinky rotten eggs, it's actually toxic for us.
Oh, yeah.
So because it blocks basically exactly that spot on the hemoglobin, and preferentially it's faster than the oxygen, no? But they have a hemoglobin involved with two docking sites. One is for the hydrogen sulfide and one is for the oxygen. And therefore they can transport both of it. And while they transport it, of course, it's not toxic for the animal because it sits on the hemoglobin. stop, and that's how they do it. And now we stop the pump, please.
Yeah, that's super interesting because I know some marine animals have that blue blood, right? Like lobsters. That have
Yeah, some crabs. Yeah.
which is
This
a
is
different kind of hemoglobin again. Huh? Which is
this is
cobalt or something.
hemoglobin, hemocyanin or something they call it. It's a different, very large molecule, but different.
Probably need to rack in, huh? Or if you
Oh,
can-
yeah, definitely right. Essential to still have the camera.
I like camera.
Yeah, it's better than not.
Camera's my friend.
Alright, we just turn the pump off first, right?
Pump off. And then we always put also in the other direction from
Back to and almost.
both of them. Yeah. So
Musst du schon essen gehen, oder? Bitte,
hast du schon gegessen?
Ich habe jetzt gerade gegessen.
Achso, dann sollte ich essen gehen, ja.
Die Collection da absammeln wahrscheinlich, gell?
Ja, die wieder mal ins Gut ein paar Menschen holen. Und dann einen Einsatzfahrer haben wir gesagt, ins Große eine. Oder ins Kleine. Ins Große
Ja,
haben wir gesagt,
da steht es im Kleinen. Ja. So, die 4 oder die 3.
die können wir in die dann die dann können wir so eingeben und da sind nur Muscheln drin und so viel rein.
Also nur die 4.
Die beste. Und vergiss nicht das Lachen, weil die Kollegen wieder mal und das Absaugt, das sagst du
Am Schluss.
Nein, ja, wenn du das abgesaugt hast,
Also zuerst
so.
sammeln.
da ist er dann fertig, und 3 und 4 Sachen sind Minuskin, braucht man nicht. Okay, aber ich bin nicht so lange brauche ich nicht.
Also was jetzt mal alles anräumen?
Das anrahmen.
Should I log out submission to the Microsoft?
Pupils and muscles. Dazu weißt du genau.
Pump is up.
Pump is
Pump
off.
is off, great.
And then you're gonna bring that, leave it on the bag, and then pull it back, so.
You can take this and store it.
Okay.
Das heißt, das Abkommen und dann den Sack einfach in den feinen Netz tun.
Genau, ja, aber bei den Rehren will man was das so gut wie es geht.
And we want this part here, right?
Yeah, at least something of it, no?
Yeah.
Like, what is that?
Waypoint
This is
13.
Control experiment 2, and then you have to make it the observation that you have to write all the speed control. What is this now? This is observation.
Bump.
Okay. Okay.
Okay, gut.
And this is
Why are you throwing stuff at me?
das Kind auf hier, jetzt wieder von dem den Mist.
Passt.
You want me to porch out there, Chris?
I don't think you can.
Just do it while doing it.
I think that he's starting to.
Das tun, das tun.
Yeah, right there.
You can always let go and do a side grab, you know? On the sit, yep. Is that enough?
Yes.
Come down. There you are. If you let it open jaw now, you should be able to just bonk her in.
Was I missed?
Get that on these big jobs, Chris.
Alright,
One now.
okay. That's in. Flip it completely.
And then we're gonna collect
Yes, so
Where
next
we were
thing
doing
we
water?
collect this part here, where we just made the temperature measurements. We put it into BioBox 4. So probably best is to try to take the scoop to loosen some muscles, and probably that's the way to start doing it.
no problem.
Very good, thank you.
Sample to sample 26.
Sorry?
Sample 26 scuba.
I think so.
Yeah, yeah. If you, or do you want me to do one of the arms? What do you want?
I just need to be able to get to the back of the
Oh,
porch.
so I have to porch out now, right?
Yeah.
All right, I'm gonna porch out, and if it bumps me a little, that's okay. I don't see anything behind me really. Porch out.
Sabina? I started the sampling. I did something. What's the result? No. And this is
That'll work for you?
Chicken control.
The tube box there up there. The tube box and the muscles up there.
That box.
It might actually be easier with the arm.
Other arm. Yeah. So I put the high temp probe in coral cover because the high temp probe and the wand for the mass spec will split like this. No way I can get it back
Okay.
in that.
Oh, okay.
Sabina?
Yeah, it looked like that holster's like gone, or?
No, it's okay.
So there-
The gray one, the one that was in, yes, it's possible to see from this angle.
Box 4 you open in the beginning or afterwards?
It doesn't really matter to us.
Because of the bungee.
I'd say after so we can see it. If that's all right, but yeah, we could go ahead and do the bungee now.
Yeah, because otherwise you have the muscles in your...
Yep, that's no problem. Do you want me to do the port arm to do that?
Oh, the bungee? Oh, we're putting it in the front ones, okay. Yeah.
If you're happy, I'll porch in a little bit.
All right, which one we put it in?
We're gonna go in 4.
All right.
I'm porching in.
See right in the front there?
Yes, sir.
this.
Very nice. If you can, put that somewhere you can get it again, because we'll use that one when you put it in there, you know?
Yeah.
Probably good there.
Don't want that too much spray. Alright.
All right. Just let me know if you want to move or get closer or anything like that.
That should be alright. We're going for muscles first?
This pile right here in the middle. Or do you, we're okay to go ahead and scoop this?
Yeah, so I would like to probably start here. Probably that's the most easiest to catch in the beginning and start losing it from here and then work towards backward.
Sure. And basically you want that whole pile?
Yes, basically where you just measure the temperature. So with this we want to have as a collection.
Okay.
If it's possible.
Got some big muscles, Chris.
Mm-hmm.
Yeah, let's straighten this out a little faster.
I can do the other arm and kind of push him in there if you want.
Might, yeah, so we'll see that. Yes.
Very good.
I can push out a little if you want.
Um,
Or you just leave it.
that should be okay.
All right, open in 4. Gold.
Alright.
So I might as well leave that open. Now. You just gonna let that-
Yeah, I don't think they're gonna fly away.
We're just picking muscles.
We got 3 or 4 in the
Nice.
right now. She wants all the muscles.
And sorry, and also the tube ones, but it's probably just easier to start with this, with the muscles, and then go to the tube ones.
The whole buffet. Event buffet.
It's lunchtime here on Falcor 2, so we got a watch change. Got Russ coming on as co-pilot on the manipulators.
Let me know if you think you could benefit.
Zach's on the pilot.
Forward arm and kind
And this is Tyler
of doing that.
in the multimedia tech spot for the moment.
Just because it's easier to see.
You want these two? These
Sorry?
as well?
No. So
No, only
only so where we measure the temperature. So basically these here and basically everything that's like in this area.
All right, let's see if we go on.
Yeah, I can move as needed or anything.
Getting close to the range.
You could pass it to the port arm. Let me see if I can hang it.
So, yeah. It's a bit close.
Yeah, that's a big clump.
Might be under port. No, it's not on the range.
Don't worry.
I can just lay down. It's pretty hard. That's cool. Poker it up, Russ.
One more. All right, and then you want me to take it or just help you put them in there?
See if you can scoop them into this.
Sure.
I can just,
That's one of them right there, is this pile, right?
yep.
All right, I'm just gonna come down on the other side of
So
this.
we're gonna work together to grab these clump of mussels.
All right.
All right,
I'm ready to,
push them towards you.
yeah. Actually, it's getting that a little bit broken up so we can see.
All right.
If you're all right with it. There's one there.
Maybe if you come in at a little more of an angle to where if I bump them in there, they'll stay. You know what I mean? Or can I grab these with the arm?
Yes.
It's okay.
So we've got one pilot on the port manipulator
Well, if I can grab them, I can.
and one on starboard manipulator with a scoop. We're trying to figure out the best method to get these mussels into the buy box.
Just don't crush it, so I'm gonna take my time.
Well, it's easier to just put it in the box like that.
Yeah, I could. I can let go here though too, I think you got it.
Yeah.
Alright. Froze, locked. Do it again, alright. What'd you think of the Turkish pie there? Not bad, huh?
Good.
Yeah.
Probably
I'll be here if you want another hand.
can try to get these two bolts here, starting from that side.
Yeah, you want to get those right now before the muscles?
Yeah, or like you can try to get this one as well before. It doesn't really matter. But,
Okay.
what is first?
Yeah, the shoe ones, we don't scoop those, right? We
No,
grab
no,
them.
you have to
Grab it back.
I don't know. You would come back.
Okay, get a question for me. I will be back in 10 minutes. This is good.
You are with the muscles, no?
Yes, and then after the tubes.
Okay, and with the grab, we need to pry in there also for the two ones. Did you go with the pry bar in it?
No, not yet. This
Okay,
is in the end.
okay, good.
We come back.
Kind of, do we know kind of an estimate of muscles?
I wrote it down there right now. 7 muscles
Okay.
in the box.
Okay, and 4.
Thank you. Yeah.
Good. Mm-hmm.
I flip these out of here, so I can, because they're all stuck together.
Come in if you want. Can we come in there?
Yes, please.
Alright, just back up a little bit. I'll grab them like last time and drop them in there. Maka, how's your lunch?
I'm stuffed full and it was very good.
Did you have the cheesecake?
I had everything.
Over. I'm gonna let go. Ready?
Six. Stuck on the back of the helmet.
The scoop is too small.
What's that?
The scoop is too small for the muscles. On top of it.
Could be too small?
The scoop is too small
Oh, the
for,
scoop is too small, yeah.
for those muscles.
Whatever.
It's okay, that's okay. We'll go.
They're right there. Here, if you want, I'll just huck them in the box after again.
Yeah, just do.
Okay.
It's kind of nice how they all stick together like this, you know?
Mm.
gonna dig another group of them here with this thing, or how you gonna do?
Well, I'll see if I can see it. Well, there's that one there. Yeah, so give it a shot. And zoom in, that'd be good.
Yep. Well,
This, and I qualify a highlight video
Okay.
for the
So it's-
scooping of the muscles. You want to keep track also of the number of muscles?
I do this. It's okay, I do that. Firebox
4.
4.
It's gonna help. I can't get it.
No problem. No, it just might not be the best. So these.
Now, I think this scoop is really good to loosen it up, no? And then maybe if they're loose, then
I got you.
Just get it straight around.
You want these right here as well, right? This bench. See where I am?
actually this is what probably is enough because,
See that guy I'm about to grab?
yeah, you can grab this guy also.
You want him?
Yeah,
Or her?
yeah. If you Oops.
Hmm?
Where?
String the bolts.
Yeah.
Is it possible to collect them without crushing?
Yeah.
Or we
If you
do
scrape
with
them
the
out towards
scoop,
me,
we
yeah.
scrape them or something?
Yeah, just bring it out towards me. I'll see if I can.
Because it's,
Okay. Yeah, the last one I did pretty good, but this is the
I
first
think
one
this is
that's,
very difficult.
yeah, that last one smooshed.
Yeah.
Let's see, maybe I could just not close, just kind of brush it over. Are these
Oh.
very delicate?
Yeah, they're not really so strong, no?
Not super strong?
No.
Maybe it helps to slide them a bit on the bottom without closing, and then only when it's off, take.
Yeah, well, see if I can scrape them out towards.
With a scoop?
Yeah,
Yeah.
it's okay if we move them.
Okay. Sure. Not closing.
Seems that the scoop.
So an inherent challenge when getting collections using these manipulators is, again, there's no tactile feedback. It's all visual. And to close the jaws on the manipulator, you have to push a button. So you have to be very delicate, and it's often difficult to get delicate organisms from the seafloor if you just have to lightly grab them. So the pilots are working together to find the best way to collect these samples with the least amount of damage, which is a considerable challenge often.
Right on the edge of my range.
Yeah, you want me to hop forward?
Yeah, please.
Yeah, no worries.
Okay, there it is. Okay, come on. Even if you just skipped over, like a little bit, put that railing line with that frame.
No worries. For a wee hop.
So the pilots are adjusting the location of the sub. to make these collections a little bit easier.
Let's see how that actually. So, obviously we just moved, just getting this clump, you know, right here.
Okay.
So that's all going in 4. It's open right now.
Okay.
And we're pushed in so that you can move the arms around a lot.
Sure.
And some, we've had some luck with just grabbing some and throwing them and some with the scoop, and just kind of doing whichever makes most sense, you know.
Sure.
Yeah, this is loose. This one is loose now.
The bungee we just set on the next box.
Okay.
So, grab that, no problem.
Well, that's how you do it. I kinda get it.
Just a little shortage happening here. You can get the tip close. I can either
Dr.
tip
Monica
it
Bright, our principal investigator, is
or you
on
can
scientific mic.
scoop under it.
Jill is our pilot. Alex is the co-pilot working the manipulator. And this is Tyler on the other mic. And if you're just tuning in, this is live footage coming from ROV Sebastian, 2.5 kilometers beneath the surface of the Pacific Ocean. We're in the Underworld of Hydrothermal Vents expedition. And today on our dive, the science team scouted out a location for the next experiment. And currently we are collecting samples of that location in preparation for deploying the experiment tomorrow.
Just slide it.
It should go to a fridge or to a
Looks like our pilots are using a bit of a dustbin method to get those muscles in the scoop working together.
So today it goes to the refrigerator in the lab.
Again, one of the challenges of doing these collections is finding the right perspective for the pilots to be able to see what is happening on the seafloor 2.5 kilometers below RV Falcor 2. So they're relying on multiple camera angles to try to judge the perspective and distance as they use those manipulators. So I've pulled up the two primary cameras they use for collections, our situation cam and the science cam. And as you can tell, the angles on both are not the clearest for making this collection.
Then you get everything good.
So the pilots are doing their best to make this work.
Das morgen ganz drauf die Windröhre wegnehmen.
Die große,
Die große und die große.
was ohne Schema steht.
Die
Ohne Schema, also nicht auf nicht auf.
genau.
Oh, ist das
Die
verschiedene
Schale,
Schema?
da die das Gewicht noch drin sind so breiter Profil.
Dann haben wir aber da irgendwas Todes. Okay, ja. Schauen wir mal.
Aber mit Taschen, so ein paar sagst du. Kannst du nicht so ganz gut.
Ja. Ich habe zuerst gerade überlegt, wir können nicht mehr tauschen, ne? Aber ich habe zuerst gerade überlegt, wenn wir da, also eine ist ausgefrischt, aber die anderen sind ziemlich gut, das zweite für die Muscheln, aber die brauchen es für früher, ne? Und wenn man die wirklich abspülen, das ist ja die richtige Aufsaugung, ne? Aber ich brauche noch keine Muscheln mehr sagen, weil ich glaube, dass die Röhren würde nicht eingehen und das vielleicht besser sind, wenn sie hinten sind. Was ich meine, aber wenn es vorhin sind, ich weiß es nicht, wie man das aufteilen werde. Ich brauche halt keine Muscheln mehr sagen, wenn ich die kriegen will. Weißt du, was ich meine? Sehr voll abspülen, so. Und das ja noch Gips da.
And another muscle in the bio box.
Weil ich glaube, das geht ziemlich raus, wenn man da mehr noch abgraben will. Das ist ja nicht wahnsinnig, dass man nicht doppelt so viel Muschel da drauf ist, was du überhaupt brauchst. Du brauchst ja so viel Community. Ne? Und dann ist die Frage, wenn man das werden will, man das wird, wenn man nicht und das anfangen, dass das auch nachgehört ist, wie schwer das ist einfach. Ja, das bin ich gerade überlegen, weil ich da gut sind brauchen. Und dann würden wir die nicht gut sein.
Und dann bringen die Winter rein, ne? Die Ansatzware.
Dann kann ich die Winter einfach da und dann habe ich aufgeschrieben, dass die feines nicht saugen. Nicht, dass ich das vergessen habe. Das sagst du ja, bin ich vielleicht schon wieder so. Aber und
Also die Muscheln nicht da rein, dann die Röhren rauskriegen mit den Steinen. Du magst einige.
die Steine kommen dazu, wo es gut sind.
Genau, aber du
Das
magst
ist mir
also
wurscht.
einige. Das man nicht
Ich
rauskriegen.
möchte einige in den Steine zum Anschauen. Ich habe nur einen gehabt, zum Beispiel. Nur einen gehabt, das war so Fläche, so Oberfläche. Ne? Also ich würde gerne mehr Steine, aber ich will ja sehen, wie die Stelle ist.
Also nur so den ganzen Stelle, dass wir das quasi nur eine kann in die zwei, in die 1, 2.
Ja. Und den Stab dazu dann einfach nur. Weil, schau mal, da hast du auch so einen Crack, aber da muss der Crack irgendwie laufen, dass da mit dieser Bar einfach da, da muss irgendwo ein Platz sein.
Ja.
Und halt, die werden wir mal hin sein, aber dann hast du die Aufsagen und selber so, das ist ja wurscht.
Und am Schluss die eine von dem
Die eine
Sollen
und
wir die
die,
einfach dort lassen oder sollen wir die wo einstecken?
die steckst dazu, das ist ja wurscht. Also, und die Muscheln nicht verschieben. Ah, nice. Das ist wieder von Massaker, aber wieder von Massaker, aber
Ja, wenn ich offen willst, dann.
ja. Ja. Ich meine, das musst einfach bei der, wenn du den Bärenlohn nicht drin haben willst, dann
Und die rennen da hinten, ne?
musst du ihn einfach abreißen. Also
Genau. Ja,
der nicht abreißen,
ja,
einfach.
aber das macht ja nichts. Ich
Da
kann
kannst
nicht abreißen.
du kannst gleich angreifen lassen, so wegreißen lassen und die Muschel halt, oder dass das andere dann verschieben sie halt morgen noch in die, aber
Nein, die Position ist
die
gut. Ich glaub,
Position
nur-
passt schon, aber
nur der eine
Aber nein.
Musik.
die wird sie verschieben, wenn es da anreißt. Ne?
Das macht
Das wurscht, das macht nichts, das mache ich, das habe ich gemacht.
Another good collection.
So it's not some message to learn that we will
Wir werden es wahrscheinlich so machen, dass wir die Muscheln wie du schmeißen sie in die Firebox und der Rest in die Big Firebox. Und ja,
you let the two go down in my notes for
whatever. 4 and 1, 2 will be the same because
See the
it's always
two
much
berms
faster.
moving away from the incoming scoop,
And
or retracting, they're not moving.
For the for Jesse.
Die sind jetzt schon drin. Also ich habe jetzt, ich glaube, das sind jetzt 10 und da wurde die gezeichnet und haben die jetzt 25 dazu quasi. Zu meiner Hand. Wie viel hast du gehabt? Ich habe 7 Wohnungen gehabt. Ja, jetzt sind es insgesamt 12. Also das und eine davon ist mein Schutz. Okay. Das 13 lässt sich mehr schon ungefähr, aber ich glaube, da kommt jetzt nur eine. Das ist jetzt dann. Das ist schade. Aber es ist nur schon. Aber jetzt aber wirklich. Goes to box number 4, and then you want to get all the cucumbers out, but then you die because it's no way to get them out alive because they're super too deep. And we put these in the large flower box and we want them. And it's like one sample for us, but we want to separate them. So they will not be enough space in the flower box for them. Also wenn die Röhren raus sind, dann sage ich es dir. So on the first day, you know, when we cleaned our first area, but there was a big mess in the end, all the cracks were also opened. And this basically destroyed our site. So we're afraid if we collect the tubes now from the frame area, that we mess up the entire area. And we stain anyway. So we now basically look to, afterwards, we have to look for more animals and like distinguish between the red colored ones and potentially new ones. But the reason is that we don't want to destroy the site.
Okay.
And that's why we now basically take a controlled collection next to it.
Okay.
That's now our control.
I see.
So the original idea was different, so we are totally right.
Okay.
Just had to change our setup.
I understand.
Dr. Sabina Gollner has joined us in the front row here.
Yeah, I'll just, so this section right here, of tube worms, we're gonna end up pulling out anyways because the roots are so buried that they're not gonna come up. Right, so I would just scoop towards them, that worked really well for that one time. And use those to help it push it onto your, you know.
One thing about being an ROV pilot is you learn the best methods to collect different deep sea organisms, and there's a trick for collecting a lot of these species. So there's a lot of inherited knowledge on the ROV team helping each other out and the best way to do these maneuvers. another muscle in the bottom box. Nice.
Very good.
Now you might watch these collections and think, oh, that's easy, but I can guarantee you that it is not. Because again, these pilots are doing this entirely with visual feedback, looking at these two different camera angles that we've got up on the screen here, and they're using a rigid tool against a hard seafloor, and there's no feedback outside of what they're looking at in the cameras. So whereas you could easily scoop something up with your hand because you have tactile feedback, you know, the pressure of it, they're doing all this through sight. And another muscle in the bottom box.
Got it. It's just a little stuck. Oh, sorry.
That's in.
Yeah. And while this hard seafloor is unforgiving when using a tool like the scoop, one benefit is we're not stirring up a lot of silt every time we touch the bottom. So the visibility is staying pretty good throughout these collections.
So do you think you have enough? We can just move them, or you want to just keep collecting?
Now, I would like want to clear this area here.
Yeah.
So, I don't know if it's best to first collect these muscles here or start now collecting with the tubes. Maybe that's easier to
I think we can keep doing muscles as long as you're
Yeah.
happy to take more.
So just want to clear basically this area. So yeah,
Okay.
you can collect these here in the back.
You can see that metal frame in the right lower part of the screen. And that is going to be the excite for tomorrow's experiment. So if you tuned in yesterday, you saw us deploy 2 boxes, fill them up with dye, and then pump the dye back out and leave one of the inner mesh boxes there. So this will be a site for a similar experiment that we will deploy tomorrow. So right now we are clearing some samples from this area. So Dr. Sabina Goldner is working with the ROV team to identify mussels and organisms she'd like collected, and they're grabbing them for her.
Also all the limpets on the ship.
No, so you can see it.
Occasionally you might see the manipulator pause for a moment, and oftentimes the pilot is re-indexing the arm. So their controls are essentially a miniature version of the manipulator here in Mission Control Room that they maneuver however they want the arm to move. So occasionally I have to freeze the arm on the seafloor so they can adjust that manipulator controller so they can get the right angle to finish their moves.
Okay, I think we're good with the muscles. I'll try to get basically these tubes here. Sorry?
The worms.
The worms, yeah. And we place these worms in the bar box 1 and 2.
So we're going to try to get them with the scoop or should
You have to, I think, pull them out. And first, yeah, they will retract a little bit. But it will be very difficult to get them out. So we do not expect that we get them out in one piece because it's not possible. So.
Dr. Sabina Goldner setting the expectations for this collection. These are difficult to collect, these tube worms. So we're going to take a little bit different approach here. Looks like we're going to put the scoop away maybe and just use the manipulators and put those worms in a different bio box.
Like for the whole dive?
Yeah.
I don't know. I
Put it in
wouldn't
that
put it
forward box just in case. You can always move it
Yeah.
later.
Mhm.
If it goes in the back, it's going to be tough.
difficult. Yeah.
Oh, sorry. Forward. I like it.
That's perfect.
It's perfect. Sounds like a little easier. But I don't know what to say. So, I'm good with that. I'm gonna read back up because that pushed me a little back.
So they're pushing out the porch, actually move the sub back a little bit. So Chill is going to adjust the position again.
There is trims on. Yeah.
So these are the most easiest to start with.
Is that good or you want to be closer? That might be good. Like I'm not super extended. So it's okay to grab kind of to the base with these
Yes.
this time? Okay.
And also in the end, we also want to crack the rocks. So you also want to test a bit for tomorrow, like not for tomorrow, but like for when we recover basically the mesh boxes, to see how difficult it is to get inside the rock.
Okay.
So first collect these tubes, and then after that we try also to get some rocks from beneath.
Okay. So are we going to try to still do a little bit before, but like scaring it back in, or just grabbing up the bottom for the base? Pushing this one's way up. What do you think, the other end, this one, is it working? It's already just launched, so. You might just be able to barely grab it and see what you can do. And it's a muscle, you know, want to grab the muscle.
It's also possible here, because I think they stick together. Probably. No.
Is it okay to grab muscle with it?
Yes, it's also yeah. Mhm. just, placing the box mounted.
Yeah, so I can't push out, it's just a grab on. I'm gonna have to come up, the animal grab on. Sorry.
Yeah, it's like still, like you can see here, the animal still that's like in the,
Let's pull the two
yes,
off the
yeah,
line.
yeah. But you have to, I think, move anyway. Yeah. Ah, here we go. With the wall.
Okay. I'm gonna have to float up to be able to push out. This is gonna be kind of a slow process here.
How do you feel about that, Alex?
You can actually see here very nicely the inside of the, what is the animal, what is in the tube.
Yes, we have maybe a rare view of a tube worm here. And because of the issue with the porch pushing the sub out, Jill's come off the bottom a little bit to porch out so we can finish this collection. So while the collection itself wasn't the most difficult, getting it in the box is a little bit of an obstacle. So from this top-down view, you can see that we are about 1 meter from the seafloor right now. Now we finish this collection. And then Jill's going to set us back down where we were before.
This is gonna take us a while. The problem is when you shoulder down, I think it's gonna take a little bit longer, but we'll make it look better. Just come out and push it down.
You cannot stay down.
Every time I do, the Porsche hits the rock, it moves. It's because I can't see, like I don't know what's dragging on the rocks. Sometimes I can do that, and sometimes I don't feel like doing it.
Next, I would try
Okay, I'm down.
this here.
So the pilots are establishing an order of operations for these collections. Now it looks like we may have to lift off the seafloor, porch out, finish the collection in the bio box, bring the porch back in, and go back down the seafloor.
Touch it first. Oh.
I think she said you gotta grab it from the base. They're so in the rocks, just no normal connect collection. Okay. I can try it one more time and see if the porch operates. That's not so bad.
So this top-down view, you can see the sub moving a little bit as the porch comes out, but appears to be all right. then forcing back in, sub moves forward again. So Sabina, once these samples are on board the ship, what will happen to them?
So different things we have with the samples. So one of the purposes is here to get to know the community, the natural community. So we look basically what kind of different species we find there. So we, for example, identify all the limpets that are there, all the polychaetes, the bristleworms, the bristle stars. And for example, with the mussels that were taken before, that we put in the front box, with these, they will also do experiments in the pressure vessel. Also the tube bombs, not these that we collect now, but afterwards, there are also experiments in the pressure vessel, the fertilization experiments that are done. And basically with the goal overall to find out under what conditions these animals can live, so under which temperature regimes and what they can do under which temperature regimes when it comes to reproduction.
And the container on the back deck, that's to simulate hydrothermal vent conditions, correct?
Yes, so the Gergis lab, they have like a full container with them, equipped with high pressure vessels, large ones, and with a flow-through system where they can regulate also the oxygen concentration, the sulfate concentration, the pH, and by that you can really study like under what conditions the animals can live. And we also have a number of, I think, like more than 10 or 15 smaller pressure vessels in the main lab, which we use then basically for the smaller animals, also to pressurize them, put them again under the conditions that they are in the deep sea, and expose them to different, again, temperature regimes, oxygen regimes, and so on. Also related to our question, when we want to know like if they can travel in the subsurface, we know the conditions are extreme there, so we also need to find out how long basically these animals can endure such extreme conditions.
Excellent, thank you. So there are a lot of ways on the ship at this point to reproduce these hydrothermal vent conditions and to test whether the samples and creatures could survive in different environments.
Which one next?
Maybe this one?
And so if you've seen pictures of Falcor 2, our recently refitted research vessel, we have a massive back deck, and currently on that back deck is a shipping container that's operating as another lab basically.
I can also grab this one first, it doesn't really matter. Probably can from this side.
And if you're just joining us on this live stream, we are putting out footage from 2.5 kilometers beneath the Pacific Ocean. And you're looking at the eyeballs, basically, of ROV Sebastian. It's tethered to research vessel Falcor 2. And we are on the Underworld of Hydrothermal Vents expedition here on the East Pacific Rise. So currently, we have so far today established a site for tomorrow's experiment, which will be similar to yesterday's. But right now we are collecting samples from close by in the area for the science team on board to do various experiments with. So right now we're collecting tube worms. A little bit ago we collected some mussels. And Dr. Sabina Gollner is on the scientific microphone. We have Chill as our pilot. Alex is a co-pilot. And this is Tyler on this microphone. And if you have any questions in the chat, let us know and we'll do our best to answer those if we can find time while running these collections.
So the last ones that we did.
Sideways, as much as you can. She's just trying to minimize the amount of contact.
It's good. Yeah.
So Sabina, we had a question on the chat. We've seen some empty mussel shells. Is that a result of predation, or why do you think that is?
Oh, it's a good question. I cannot directly answer it because there are basically two different options. The one is predation, and you can also see crabs, but I think it's actually rather unlikely that are predated. I rather think that this is the remaining of a dead mussel, so that just died because it was old or there was no vent fluid anymore. And then the body would, basically degrade over time. And we have been in this area here before, and once we observed like a mussel patch that had no vent fluid emissions anymore, and we could see that the mussel shells dissolved within 2 to 3 years. So if we see here an empty shell, it's rather young. So imagine a mussel dies, the shells are open, then I guess that the meat is like predated, and it's already dead, the mussel, by all these crabs that are around. And then the empty mussel shell remains, and this would dissolve within 2 to 3 years.
All right.
Mm-hmm.
Meet the chisel.
First, we need to take. I can show you in a second. So, if I look at the frame, There's one thing that we're worried about for tomorrow's dive. That probably will be difficult to put the box on top. So if you could remove this one tube one,
Okay.
that would be great. I can also go to bar box 1, 2. And the mussel can be either put also outside or so basically these two specimens.
So inside the frame area, you see that tube worm on the left side?
Yep.
That one needs to be removed. So we'll probably have to remove the frame, collect it, and then put the frame back. That, and there's a muscle with the two, so both of those.
Dead center of the picture is a metal frame that's going to serve as the site for experiment 2, which will be deployed tomorrow. So what we're going to do now is clean that site up just a little bit. So on the left of that frame you can see a tube worm and a mussel. So we're going to remove those to make the site more manageable tomorrow.
Yeah, so leave the frame.
Yeah, I can leave the frame.
When we deploy,
We'll
it's a length experiment.
grab these.
Yeah, that's fine, but you don't have to, I think, do it away. You can't if you think it's easier, but you can I'll probably first move away the muscle a bit. Click and 4.
And we're going to collect this mussel.
No, it's not.
Actually, I think probably it's not necessary to take the tube on because it's also miss the camera. These, when you put it on top, also with the weight will push it down, I think, a bit. So it should be, because when we have like this mesh box, also inside there's some weights mounted to it. So actually the surface area is a bit smaller than the frame. So that's why the mask is 2 bits away. So I'm wondering if it's worth or needed to take this tube out. We leave it in. Okay.
And
Next
it's been
one is
decided
the-
that we'll leave that tube worm in there without having to remove it.
Okay, now we need to slurp the area that cleared first with the fine mesh, so suction sample 1, this one. We'll just remove all the muscles in the tube worms. basically, yeah, the spot here. It's in 1, 2, and 3. Can you rotate from 1 to 4? Is this a problem? No. Oh, okay. So start with the 1.
6.
Among the many tools and sensors that ROV Sebastian can carry down to the seafloor is a suction sampler.
Sabina, this is an example.
So currently the plan will be to use the suction sampler to, in rough terms, do some vacuuming on the seafloor.
27.
So you'll see a tube deployed,
The jar.
it'll suck in
Try one, but we have to
organisms or whatever else the scientists want, and we have a multi-chambered container on RV Sebastian with different levels of mesh depending on how fine of samples they want.
Dual breath control.
Sorry?
Dual breath control is something.
Almost need to check Zach if he did 2 and 3.
So this is a view of the suction sampler containers, jars. They're all numbered. We do have a camera pointed at them on the ROV so the pilots could check which one is lined up. And we're just conferring a little bit to determine which is the proper suction jar to put these samples in.
Number 3 also is a small mesh, so 3 is easier. You can also take the number 3.
So another quick update for you if you're just joining the live stream. This is live footage from ROV Sebastian, a remotely operated vehicle. It's currently 2.5 kilometers beneath the surface of the Pacific Ocean on these Pacific rides. So this is the Underworld of Hydrothermal vents expedition, where we're exploring a very interesting and unique idea that organisms in these vent systems, particularly tube worms, may be able to travel beneath the vents to repopulate different areas. So today we've set up a place where we'll perform an experiment tomorrow. We've done a bunch of sampling. So right now we're getting our suction sampler prepared to continue sampling in preparation for our experiment tomorrow. Now, when these samples come back on board, the scientists have a variety of lab spaces set up that can replicate hydrothermal vent conditions or even more extreme conditions, so that they can examine these animals and see what conditions they can live and thrive in, among many other experiments.
Is that a crack in there?
Oh, yeah.
You see that?
Yeah. It's probably me just now.
I know, it's like a bolt. Can you go the other way around where you're trying to? So we're trying to get to 1, 2, or 3 basically.
So Sabina, we had a question. Are there any animals that feed on tube worms?
So I do not think so. So what we have observed is, or when you look at the tube worm, it's sitting in the tube. The animal is sticking out, so there's it's protected in the tube, so it's difficult for predators to feed on it. You see that sometimes these Butoquea crabs, these white crabs, try to get some pieces out of the Riftia tube, of the plume. But I think other than that, no.
All right. So not much predation happening on those tube worms, and if you've been watching, you can see the red plume. When we touch it, it retracts back into the tube, so it's a bit of a protective maneuver, I guess. But thank you for the question, and if you have any other questions, feel free to send those into the chat, and we'll do our best to answer them. And while we got our suction sampler set up, got a little bit of view of the ocean surrounding Falcor 2. So we've had a bit of weather the past few days, nothing too terrible, but it's been cloudy, but today we've got some nice clouds. A little bit of roll on the ship, but things are feeling pretty nice here today on the East Pacific Rise.
Okay, which one of those when he does it? You want to grab the suction sampler?
One of both the internal groups, you can see, because he's looking at adjusting the valve. The unicorn or the index?
Unicorn? Okay. Alright, this is not one of these. Should be off that starter valve back. I don't see it. Alright. Yeah, they were just asking if we could get any more out of it, and I said I'll check the valve numbers, but. Put a clock on it. Yeah. Oh, it's weird. Alright, it's not what it's on.
I think you tap this, what happened to that? You gotta, like,
So while we're paused, Sabina, just think you could tell our audience how you arrived. What's your background? How did
Sorry?
you get here?
How did I get here? Ooh,
Yeah.
that's a long story. Like related to this project or in general, or what is that? I don't understand the question probably fully.
Just your background. What led you to become a scientist?
So I originally come from Austria, which is, as you know, not situated at the sea, but I was always interested in the ocean and life in general. Then I started studying biology at the University of Vienna, and actually ended up in Monica Bright's lab as a master's student, PhD student. And Monica then took me already during my master studies, also actually to this place where we are right now. So my first expedition was in, I think, 2003 here to this site, and then with the submersible Elvin. So it's really nice to be here again to see this. And then I started actually studying the deep sea hydrothermal vents in this area. Did my master thesis there, also my PhD thesis. During my PhD thesis was a volcanic eruption, which caused that I had to actually reshift my whole planning. So originally I wanted to study food webs here, but then volcanic eruption halved the entire area. So I shifted my PhD project to study the succession of this community. And yeah, after my PhD, I moved to Germany to their postdoc, and since 10 years now I live in the Netherlands. And yeah, I think I'm very lucky that I'm able to study this deep sea hydrothermal vents, but also other deep sea ecosystems. It's a very, very nice job.
Yeah, you've been on this site multiple times, and this is sort of a familiar part of your career right here.
Tapi the section sampler jar free.
Yes. Mm-hmm. It's jar free.
It does not look like it's on right up. Yeah, so it won't rotate.
And the
So
mesh
you're
is
gonna
fine,
have to
or-
use
It's
the
fine.
pitch of the arm to change the angle. Once you pitch enough, then you will be able to rotate it.
Oh, your HT probes wrapped around it. That's why. Nice with you.
Okay.
Yeah, it's-
Just there, exactly right there. Grab the green. Okay, got it. Yep.
Hola, buenas tardes, buenos días o buenas noches, de donde sea que se estén conectando y nos estén acompañando acá en el live stream de esta expedición del Falcor 2. Gracias a Schmidt Ocean Institute. En este momento vamos a tener un espacio en español para que toda la audiencia de habla hispana pueda saber qué es lo que estamos haciendo, mandar sus preguntas en español o en el idioma que deseen para aclararlas por acá. En este momento lo que estamos viendo es el setup para tomar muestras con este tubo que vemos acá, que es para succionar. Lo que vamos a succionar es agua, y estas muestras de agua van a ir a unos contenedores que tienen filtros para poder seleccionar de esta manera fauna que tenga un tamaño específico y así utilizar esta fauna en diferentes experimentos acá a bordo del barco Falcor 2. Yo soy Ivonne Rodríguez Ramírez, estudiante de la Universidad de Costa Rica, y les voy a estar acompañando por la siguiente hora.
Does that, knocks that out of your head. Just trying to get it out of its way.
En este momento lo que se está intentando es desenredar los equipos, sacar este cilindro que vemos acá metálico es para poder medir la temperatura. Entonces lo estamos moviendo a un lado para poder empezar con la toma de la muestra de succión.
You're in there. Loosen up a little bit more. Drop. Easy. Okay. Fortunately, I can probably help you get you moved that low. Sorry, I know you were about to get it, but try it again. No, that's good. That's not that bad. Sorry. So instead of grabbing it straight on, now that you have an option, yeah, do that. You'll be better off. Yes, sir. Are you guys doing that?
Y listo. Ahora sí se va a empezar a tomar la muestra.
Still has a filter out, it must be between particles. Oh, it's going. It's just hard to tell. Yeah, I don't think they're showing that one, but it shows it on this one.
En el chat tenemos una pregunta sobre qué son esos tubos blancos que se ven en las rocas, si son animales o si son plantas. Estos tubos que vemos acá son animales, son los tubos de unos gusanos poliquetos. Este gusano de estos tubitos es de la familia Serpulidae y se pueden ver en toda esta zona, es una comunidad muy abundante. Acá en el fondo marino no se tienen plantas, de hecho, y eso es porque en estas profundidades no llega la luz, entonces las plantas no pueden hacer fotosíntesis. La luz que vemos en este momento es gracias a luces LED que tiene el ROV instaladas.
También se ven filamentillos blancos que son bacterias vegetativas que hacen la oxidación del azufre. Las partes que se ven lechosas en la grieta pueden ser también lechos de bacterias del azufre, de vida libre.
You want me to stop the photos?
No, leave it. I'm just signing off, just shaking my fist. My output, so that stuff like this.
En este momento, liderando la expedición, se encuentra con nosotros la doctora Sabine. Por si quieren hacer preguntas específicas, ella es una de las investigadoras principales de esta expedición. Sabine, do you want to explain to the audience what are we using these samples for?
So we want to understand what communities lived in, or to be able to compare it to the subsurface community. So it's actually our control sample.
La doctora lo que nos acaba de mencionar Lo que nos acaba de mencionar acá es que todas estas muestras son muestras de control para poder comparar las comunidades que se encuentran en el ambiente con las comunidades de organismos que se encuentran asociados a los tubos de los gusanos Riftia. Estos gusanos Riftia son los que podemos ver en pantalla, que tienen un tubo blanco grueso, y en el extremo superior se sale la pluma de las quetas de este gusano, que son color rojo.
I think part of that is that fine mesh. Really slowed it down, but I could be wrong.
También se va esta colección en la superficie va a servir para compararla con la otra colección que se va a tomar de material debajo de las piedras, que se quieren comparar a nivel de bacterias. También vamos a estar trabajando con los virus y con los animales, la comparación de las comunidades que están en la superficie con las que están por debajo del suelo.
Que recordemos que el objetivo de esta expedición es justamente ver cuáles animales utilizan las ventilas hidrotermales para movilización. Específicamente validar esta hipótesis de que los animales y organismos microscópicos utilizan las ventilas como una manera de movimiento de un sitio a otro. Ayer justamente se instaló un experimento para validar esta hipótesis, uno de los experimentos más complejos que se han instalado en la historia de la investigación del fondo marino. El experimento se basa en tener una caja que tiene en cada uno de sus caras una malla de 32 micras. Esta caja se pone sobre una de las grietas de estas rocas. Se pone un tinte para poder tener los animales que están presentes ahí en ese momento y se deja por una semana. Luego volveremos al sitio, colectaremos los animales que estén dentro de la caja y los animales que no estén teñidos de color rojo, que es el color del tinte, van a ser los animales que llegaron a ese punto por medio del flujo del agua que salía de esta grieta de la ventila hidrotermal.
Yes.
No, probably also the brittle stars will go in. Ah, yeah, look. Muscles.
En esta zona hay una alta diversidad de organismos. En este momento podemos ver varias almejas, estrellas quebradizas, lapas que son estos moluscos de una sola concha en forma como de sombrero asiático que están pegados al sedimento de forma ovalada. Varios poliquetos pequeños también vemos por ahí. Una gran variedad de animales. Además de estudiar la fauna de esta zona, también a bordo del Falcor 2 se está haciendo experimentos con comunidades microbianas, también con suspensión de partículas en el agua. El experimento con las comunidades microbianas es muy interesante porque para ello las muestras que se toman son de rocas. Se toman rocas pequeñas que a bordo del barco se pulverizan para luego hacer análisis genéticos y ver cuáles bacterias viven dentro de los poros de estas rocas.
and put this one here, and then we switch to the finish.
Switch it now?
To the large one, so.
No.
Yes, you can switch one.
Stop your flow.
Para las personas que se acaban de conectar, saludos. Estamos acá al borde del Farco 2 en medio de una expedición en ventilas hidrotermales en la cordillera Albatross del Pacífico. Estamos cerca de un sitio que se llama Tica Field. Estamos un poco al norte. Y en este momento se están tomando muestras con un succionador, parte del equipo del ROV Sebastian, y estas muestras de agua se van a utilizar como control para compararlas con las muestras que se van a tomar en un experimento que se instaló ayer, para comparar las comunidades de microorganismos que están en el experimento y las comunidades del ambiente exterior a este. En la sala de controles nos acompaña una de las investigadoras principales de la expedición, así que siéntase en libertad de mandar sus preguntas o comentarios en el chat. Puede ser en español o en inglés, en el idioma que se sientan cómodos.
Yeah, suction, now we take the rocks, but it's very difficult to see where the crack is, if it's like it was here, but it's all a bit starting to crumble.
Acá en pantalla se puede ver, se pudo ver, aquí se acaba de salir del cuadro, un rectángulo, un marco de metal. Ese es el sitio que ha sido elegido y marcado para instalar el experimento mañana. Ayer se instaló un experimento y mañana se va a instalar otra réplica. En total se tiene planeado instalar 3 experimentos. Escoger los sitios para estos experimentos es más complejo de lo que parece. Se necesita un sitio donde la grieta no sea tan gruesa y un sitio plano para que el experimento no se caiga. Es difícil notar las inclinaciones de las rocas por medio de la cámara, ya que la cámara quita la perspectiva 3D del ecosistema que estamos viendo.
Sie gehen nicht rauf, die Muscheln bleiben irgendwo dann hängen. Die Muscheln, die großen bleiben irgendwo hängen, die gehen nicht rauf.
Normalmente se toma gran parte de un buceo para escoger un sitio apto para poner el experimento y luego en otro buceo posterior se instala dicho experimento. La instalación de los experimentos dura alrededor de 6 horas. Así que los días que se instalan experimentos nos concentramos solamente en eso, ya que es una tarea bastante difícil y necesita toda la concentración tanto del equipo científico como de los pilotos del ROV Sebastian.
wirst das Gefühl, da geht gar nichts rein.
Mhm.
Das ist voll geil. Das ist wie
Ja.
herum und dann fällt das wieder raus. Was ist jetzt da irgendwie? Ist das jetzt die 6 am Pfeil kommt? Die 6 ist das Pfeil gewesen oder was?
En la expedición del día de hoy, además de estas muestras que estamos tomando ahorita, se han tomado muestras de almejas y de gusanos riftia. Esto para otros experimentos que se van a hacer a bordo del Falcor 2. Una vez que los especímenes llegan a superficie, lo primero que hace el equipo científico es lavarlos y ponerlos en agua fría. Todo esto se hace con agua marina fría filtrada. Luego de esto, los especímenes se reparten en cada uno de los grupos de investigación para los diferentes experimentos que se realizan a bordo.
I'm just trying to find the sweet spot on the jar. There it is.
Okay, jetzt jetzt. I want like a lot.
So,
Ja?
also die Steine, so die Rocks.
Ich kann nicht schreiben, wenn ich nicht sitze. Ich kann nicht schreiben am Computer, was?
Some to 7 was fine by chance, and the sample to the 8 was large process.
Du hast praktisch aber ziemlich gut abgerannt. Schreibst du dann nicht die Zahlen noch, die kann er noch weiter sagen. Ist wurscht.
26.
Bin ich ja, das sind
26 for tuber muscle. But it's one sample amount because it's like one collection. So that's in bio box 4 and in bio box 1 and 2.
1 and 2.
1 and 2 we put the tubes.
Okay, das war nicht. Okay, gut von. Ich weiß schon, dass. Ja. Und 26 ist. 27 ist.
Suction.
Ah, du hast praktisch in beide das gleiche eingegeben, okay.
Ja.
Okay,
Also
gut.
von, also
Ja, ich meine, das kann man noch auch zusammenführen, aber es ist wurscht.
genau.
Cada una de las muestras colectadas en cada uno de los buceos es numerada y guardada en un contenedor identificado. Los científicos a bordo tienen un sistema computacional diseñado específicamente para esta expedición, en donde ingresan cada una de la información para cada una de las muestras. De esta manera se tiene un control de absolutamente todo lo que se colecta, asociada a la información que toma el ROV de temperatura, presión, salinidad y otras características químicas del agua.
Dann bei den Muscheln. Und da sind die Neiner, werden praktisch die Stauden irgendwo hinschmeißen. Die haben wir noch nicht, was schon, und die tut man. Okay. Okay, super. So, das geht dir. Okay. So, we put away the suction center and
En este momento el equipo de científicas líderes está consultando el plan del buceo del día de hoy para saber qué es lo que toca colectar a continuación. Cada día el equipo científico tiene una reunión en la que los diferentes grupos de científicos de cada experimento dicen qué es lo que necesitan, cuántas muestras ocupan, de qué ocupan muestras, y de esa manera se coordina el buceo del día siguiente. Así se puede asegurar que se colecta justo lo necesario para cada uno de los experimentos y así nada se desperdicia.
Is this locked also? I mean, das mal Muschel. Is this, did you make an observation that one muscle from the experimental site 2 was collected into?
I was just drop it.
Okay.
Okay.
Huh?
Nothing.
Yes.
All good. Okay.
This is Tica, ne? Tica, gell? Ist weg.
Habe ich gekriegt, also die Muschel.
Die Muschel ist weg vom Tito, ne?
Ja.
Okay.
Acá se puede ver mucho más claro el marco que comentaba anteriormente. Este va a ser el sitio en donde mañana se va a instalar la otra réplica del experimento.
In what observation, no?
Si se conectaron hace poco, les comento de lo que se trata el experimento. La idea es validar la hipótesis de que organismos utilizan el flujo de las ventilas hidrotermales para movilización.
And
Esto se hace al poner una caja con una malla de 32 micrones encima de la ventila y captar todos los animales que se
there
fluyen por ahí.
must be somewhere between this and this, there must be somewhere here a kind of hidden crack.
You can still see that there's one still sitting inside.
Hmm?
If you zoom in
Yeah.
afterwards, I can
Yeah, I will zoom in here, then I will look, because right now because the way we collected this.
Lo que estamos viendo en pantalla es la plataforma que tiene el ROV Sebastian con las cajas a las que se le denominan biobox, y estos son los contenedores en donde se ponen las muestras. Esta plataforma se puede retraer y extender.
Yeah.
Se extiende para poder poner las muestras de una manera más sencilla y se retrae para que la cámara.
I can. Also, this is nothing from below. No, so das ist leider noch. It will be not good. So the flaking off of the surface was happening. Just collecting or what?
You just do something like that.
Aber das, okay. But before it looked not so. Okay.
Not so
much, okay.
It's just the surface. But,
Okay.
encuentra perfecta. Por vídeo, hay de vídeo.
Wait, this takes longer. I need to look first if I see something that is.
Or use it with the other one is probably a better idea.
No, I'll try with this one just so I don't have to pass it first.
Can I focus to this place to look a little bit with the camera once you have the.
En este momento en pantalla, al fondo, podemos ver una caja. Eso es justamente el experimento que se instaló el día de ayer. En este momento estamos teniendo un cambio de herramientas. A veces esto puede ser un poco complicado justamente por la pérdida de la profundidad al tener que ver a través de una cámara. Lo bueno que tiene el ROV Sebastian es que pasa conectado siempre a el barco y se utiliza microfibra para que la señal sea rápida y casi en vivo. Justamente ayer uno de los pilotos estaba comentando de que si el ROV fuese inalámbrico, se tendría un lag entre la imagen que ven los pilotos y lo que está haciendo el ROV. Por eso es que el ROV Sebastian debe estar siempre conectado al barco. El cable que se conecta con el ROV es de más de 4 kilómetros de longitud.
He's down inside. This is good, he's not this much.
This is for rock sampling. En este momento lo que se está haciendo es romper un poco la roca del fondo para poder tomar muestras pequeñas, trozos pequeños de estos basaltos, para traerlos a superficie y que sean parte de un experimento de una estudiante del MIT que se llama Avery. Ella lo que hace es agarrar estos trozos de roca, los macera y luego se los lleva a tierra firme, a su laboratorio, para hacer pruebas de ADN y ver las comunidades de bacterias que viven dentro de los poros de estas rocas.
Maybe you can try to go below this mussel. I cannot see quite good now, but the crack is right here, and you go in right in here, below, just below the mussel. Here. To go right in here and to see if you can find this crack here, or if it's maybe really diffuse, but because between here and here is the crack somewhere, no? Because it's like it was a line that follows like this, goes over there.
Se intentan agarrar trozos de roca que estén cerca de las grietas de estos basaltos por donde se da la ventila hidrotermal, o sea, por donde fluye agua caliente.
Continue to see there, you can see it right there, and then maybe a little bit too. But yeah,
Yeah, but I would like to open the crack that goes from here, not below, but from here.
Así que en este momento la investigadora
Somewhere through
principal,
here.
Mónica Bright, le está dando indicaciones al equipo de
I don't
pilotos
think so, because if it would
para que
continue here, then
conecten
it
las
would be
rocas
somewhere here,
justamente
and
del
it's
sitio en el
here.
que se necesita.
There's something going through here, and if you go, must be because it comes up here. Maybe we can try to zoom in. But can I try to zoom in a little bit? This up there.
You can see the flow. And here was a. So here, yes, there was a thing so, but then here you can see the flow coming up. And here there was like here's the Palvinetta sitting, and we had like also one tube on here.
En este momento, si ven el flujo de agua en pantalla, se ve un poco distorsionado. Se ven como ondas. Esto es justamente el flujo de agua que está saliendo de las grietas. Esta agua es mucho más caliente y tiene alta concentración de azufre. Podemos ver en este momento una estrella quebradiza encima de una almeja. En la parte superior derecha, justo había un poliqueto.
Maybe it's very diffuse here coming up because it's
Se hizo
not
este
so-
acercamiento para poder determinar justamente dónde es que está la grieta y poder saber en dónde es que se van a colectar las rocas.
Well, these were not here. No, this is not. They're just laying around here, or is they coming up here? You see in this picture. No, no, really. Yeah, it must be on the picture because it is here, but it's not here. So what I mean is like this.
This view helps, it's kind of more from the top.
Yeah.
I think there's several cracks there.
I think, yeah, I think it's not like what we had in the other place where there's like one relatively big crack, but there's like just a diffuse area, many sort of small, no? I mean, I for sure. I wonder if you've been immediately, or were it like easy to, did you pull him out, or how much lower? So you would expect basically the thing goes like more like this, no? Because the area was originally like this. So I mean, you point here, but you know, you're pointing more in this direction, but I point more like on this line, no? So we took this away, so you're too low.
Para contar un poco sobre el sitio en el que estamos, el nombre tiene una historia muy curiosa. Estamos en este momento en Tica Field y por si no sabían, a los costarricenses se les dice ticos y ticas. Así que ese sitio fue nombrado, de hecho, por una investigadora costarricense.
Oh, I'm sorry, I thought you guys were saying this.
Okay, so
Y le puso el nombre Tica Field justamente por su nacionalidad. Cuando bajó en el sumergible Alvin y descubrieron estas formaciones, no sabía qué cómo ponerle. Y lo que se le ocurrió fue, bueno, yo soy Tica, así que Tica Field. Y así se quedó. Este sitio ha sido ampliamente estudiado.
I mean, I'm thinking more not about today, because I will check it, of course, but I think about this year then afterwards, how to get into this. No? So I mean, if I see something like this, I mean, we don't do this now because otherwise we disturb this side, but I would go like right in here where the tubes come out. And that's why I was thinking, if we find a place here, just to see it, you know, to get a better idea of what we do then the other day here, to just shift the rock. I mean, we scoop up this here first, or try maybe if it's possible to scoop a little bit of or scoop basically the rocks or the little pieces, because I will look at them, how they look, and then shift this mussel away and see where is the tube worm ending, no? Because I mean, if I would just go in here, you know.
La investigadora principal, Monica Bright, acaba de dar sus indicaciones a los pilotos. Debe limpiar un poco el sitio. Entonces van a intentar colectar ya sea riftia o almejas. A veces las colectas lo hacen con el brazo mecánico del ROV, solo agarrando el espécimen. pero también otras veces se hace con esta herramienta que vemos acá. Funciona como una palita y básicamente solo se pasa por el sedimento para agarrar los organismos que estén sobre este.
was thinking about just sliding that away, yeah. While I have a scooper.
You see? Now I can see that it must come out here.
Okay, so grab some rocks
We
here.
basically grab these rocks first, all these pieces, whatever you see here on the side you cleared before.
Acá se puede ver claro el flujo del agua saliendo de las grietas en el sedimento. Así que se van a tomar rocas de la sección que acaba de ser limpiada, de la que acaban de mover la almeja.
I think that a little bit.
Huh?
Okay, where are we putting this?
4.
Isn't, oh,
There's
okay,
the
I
mountain
thought the
cement, but
muscles.
we fold on top of it. This is just little.
Should I give us a plan?
Yeah.
Additional text?
Rocks, little rocks.
Acá entonces ya tenemos la colecta de las rocas para los estudios microbiológicos. Se toman rocas pequeñas porque son más fáciles de pulverizar. Se vio en buceos pasados que con rocas grandes a pesar de ser quebradizas, no eran tan fáciles de hacer en polvo. Y bueno, con esto termina la hora de narración del live en español. Yo soy Iván Rodríguez Ramírez. Fue un gusto acompañarles durante esta hora. Vamos a tratar de tener una hora al día de narración en español, ya sea yo o si no también otra persona del grupo de científicos que es de habla hispana. Así que esto sigue. Volvemos a la narración en inglés. Muchas gracias.
This is very difficult.
This is cool here. So there is so we broke it apart a little bit, so it's hard to see that way. This crack right here, you see everything's going in, runs into this crack here. So this line where we want to break up rocks and collect and put into biologs.
Excellent.
And it's
Quite hard.
super fun.
Good luck. Right. So you go back in and see what it scoops like.
So we got a watch change happening. Dr. Monica Bright is on the science mic still.
And I was thinking maybe you can try to pull it a little bit together.
Chris is on pilot. Russ is our co-pilot, and this is Tyler again.
What is loose already, but there was quite some little rocks loose. So it feels like that everything is kind of moving, but I don't know.
Currently, the pilots are trying to remove some loose rubble, see if there's a crack beneath it.
This is good. This is really good, something like this, yeah.
See if I can.
What's that?
Just trying to get straight in the box.
Right.
4.
So an observation.
This is already the same sample now. This is the next number, and it goes also in forest and rocks.
I have the sample.
So 29 now is the rocks.
Yes.
Good, very good. Super.
People like it that big, but it's gonna fall out.
Yeah, that's not. Just it's a bad angle. Try not to skip.
You go back in?
Yeah. Try and
That bigger one we can take later, grab it, I think. Get here, yeah.
send it back out. Send it back in. This is good. Go ahead. This one.
Yeah, but I think this is good. Yeah.
Like more.
Yeah, if possible.
Anyways, we'll just keep it straight.
The whole thing is crumbly, I think.
Yeah,
No, it's not like,
There's no hard piece, but we're trying to get straight over here.
yeah.
Gravity straight down from the top, easy to sleep.
Yeah.
Sleep from the top. So it's like doing this, doing this. You know what I'm saying? It's thought. What you want.
I'll give it a shot. That's not gonna be super easy, yeah.
So if you're just joining us on this live stream, this is live footage from 2.5 kilometers below the surface of the Pacific Ocean from ROV Sebastian, Schmidt Ocean Institute's remotely operated vehicle. So we're on the Underworld of Hydrothermal Events expedition here today. And what we've done so far today is we've set up a location for an experiment that will be deployed tomorrow. On the right side of your screen you can see a metal frame that will be the experiment site. And so we've been spending a good portion of this dive collecting samples and specimens from nearby tomorrow's experimental site. So we collected some mussels and tube worms, used a suction sampler to clean off an area, and now we're trying to collect loose rock samples from the seafloor using this scoop.
Can you get another arm and just knock it?
Yeah, no problem. And actually, I grab it.
You've already got a bar in your arm. Just push against it. All frozen.
So now we have both pilots coordinating to maneuver that scoop a little bit better.
Ideally, what I need is to push it up into the.
How do you want me to push it down so it goes straight?
If you can do that, try that. Yeah. And then go inside and pull up. So it locks into the center job. Okay.
Oops.
I just push it on the ground. She goes straight into the ground.
So we mentioned before how the manipulator is gripping a tool can make or break how easy it is to collect samples.
Oh, okay.
Pilots are moving the sub a little bit closer to that sample area to make it easier to scoop some of the small rocks out.
Mm.
In your jaw,
Problem is that
long ways,
you do that, it's no pellets.
and if you grab it, you have the bar going down, grab that in your jaw, just like a T-handle, halfway T-handle.
Yeah, you'd have to do the bar on the ground and in your hand. So you won't be able to keep it straight on the
What do you want to do?
the bar on the ground. Just push it into the soft soil. Then you can grab my strip. That's not bad. You can just try and break up some more. Off the ground.
And is this a little shoulder here on this side? I mean, can you see this? Because it looks as if this goes down here. Schau mal, this here. Because, yeah, maybe if you can try to go in there straight and then lift it. I don't know what it does because it seems to be, the rock seems to be very crumbly here. It's not like in big pieces, no. But because just to go right in, there's a tube in there, this dead already, right in there, and then lift this area. Yeah.
It does with as far down.
Okay.
Hmm?
I need to fly away with the rest.
Yeah.
Push it around. Here you go. Alright. Go for it.
Yeah, like this, yes. And as deep as you can, and then pull it back. This is moving. This is good. This is good. See? You were in there, the whole thing was moving first,
Yeah,
no?
see it. Pretty large piece though.
Yeah, this is good.
You can push under the,
Yeah, okay.
under the frame.
Yeah, we need to get it out.
Go, get off this frame. Alright, I'll get it back. Nah, it's the one crush that.
Trying to go to the side of the hole. Yeah, hold it.
Alright. I know I'm down, but if you keep doing that, it's gonna push me
Not
back
up.
up. So no, we're gonna go in.
Yeah, it's a really big slug though. I can move it, but it's moving the vehicle too much.
Okay.
Yeah, just keep moving the vehicle.
Mm-hmm.
See if we can.
Question now is because it's about, I don't know how deep it is, but would it be maybe possible because you loosened it that you grab it? Or
I'm gonna try.
also grab it with the, I don't know if this is possible, if it'll all crush, but I like to go from here, you know, to grab it sort of here
Yeah, I'll use that arm.
with the arm.
Try to screw. Really, it's screw will be good at that. No, I'm not gonna be able to get it out.
Okay.
Unfortunately. Not enough for police.
Or the question is if you would grab the two worms, if you could lift some up of that rock,
Oh, we can try it.
like just,
Which one we like?
no, not one, but just like try to grab those here and just lift it. I don't know. We need to find a way to collect later, so I'm just playing with ideas. I don't know.
Okay. As low as we can. Okay.
It's probably not working. Show me how deep they are, how they come out. There's a hole here because you took out now the lower part of the tube. You pulled out is basically sticking in a crack, no?
Yeah.
It has to be in a crack and not so small because otherwise they couldn't grow in there. And this is where a hole is.
Yeah.
So,
We'll try and work in the back there now.
no, but it's just, you can put them on the side or so. It's just, the idea would be if we can crack this rock here, because here's the crack where the tube worm came out. If you could be able somehow to crack the rock here
Well,
and
we can-
take this piece, no?
We can try with the other arm, we can just try and chip away there.
For example,
Yeah.
and these tube worms you can lay aside or so.
Okay.
I was just wondering where are they really, no? And this is actually when you see. Can you hold on a second?
Yeah.
Salva, can you focus, also can you take a highlight grab so I know exactly how deep this was here? Frame grab. Yeah. This whole thing was below. You have?
Yeah.
Okay. Thank you. I'm just basically what I'm trying to figure out how we collect the real sample then that is really important and stained.
Try
This
something.
is,
Find out this is.
so is it possible that you go like really flat in it, like this, with this
I'm gonna
arm?
try,
Okay.
just that obviously geography is.
Okay, I'm just,
No, no, you're good. See that here.
You know.
Just, see if I can get underneath this edge. Just look at it hard. It's just so close to it. It's so.
yeah.
Yeah, I've got an idea. I'm gonna use the other arm to see if we can lever it up.
It's right here. This corner is right here.
Yeah, I'm trying to crack it. See if I can
Ah,
break
you
it
have to
small
crack
pieces.
it off.
Just see
Okay.
if it'll work. If it's not. Yeah, it's not like anything else.
One, if you are able to lift a little bit, no? Because it's all moving. Go in there and then, I don't know. But, see?
Sorry, Chris, I'm trying to.
I'm trying to keep you in station. So.
Okay, I move you.
Okay. See if I have this thing again, 'cause this is not one bad.
I'll figure out a way to get this down harder.
And I really need to cut it.
Yeah, I got that. Do that. It's just for some reason very.
You can,
It's just ignore the line. That's the thing. I don't want to let it that either. Let's see if I can. No, it does not let it.
It's hard as you can.
Stand by. Let's go 30 meter off this. Then. If we get in there, if we move that. We'll move that frame, I can get further to the right.
Such a big jump. Can we work this crack here and try to get some of the material off?
You want to try to lift it from the other side?
Just trying to see if I can get a grip. I
Okay.
can't do anything on this side.
Okay.
Just see if I can get in here.
But here, I think here. Yeah, here's a crack. Here's a crack.
See, got the thing moving.
Yeah.
It's a big slab though. Yeah. Let's just work it. Let's just work it and see if we can loosen it right up. Yeah, it's gay.
Come down a little. Okay. Is the rock closest to us is the bottom.
Give us a sec, 'cause we've got this crap here. You see that? Okay.
What?
Ah, what's that? Is it here again? Okay, see it. Can you zoom me out, please?
This is
It's a masterpiece, like, and I don't know what we're doing.
the question is also if it's moving, but how would it
I
break
was trying to
somewhere?
get it lifted, but I think
Yeah.
it's too thick for us to get
Yeah,
it yet,
yeah.
unfortunately.
Okay.
Options. But we need to grab that scoop anyway.
Yeah, I'm just trying to get a breakfast up there.
Yeah.
Well, at the very least, we can get a couple more scoops of the little rock.
Yeah. Yeah.
See this stuff that's broken up here? Are any of these larger pieces useful for you? Or you don't?
Yeah, you can take some of those also, at least I can look at them.
Exactly. They'll be easy enough to pick up.
Yeah. This is just also to get an
New sample number, same sample number.
same sample number. It's just the area all. I just need to get a feeling how to look at it.
Take a periodical observation.
No, just this is all.
Back in 4 as well.
Yeah, make a observation just that.
One thing about it.
Handstands.
Yeah,
Can you move the arm, the starboard arm, please?
Yes, I can.
Because it's I'm sticking in the ground there.
Just do that.
this here.
Just, there you go.
I'll move it right like that.
Yeah.
Right.
Yeah, I was.
I know where you see it.
Yeah.
You don't know. Put that for you.
And somewhere here.
Yeah, it's right in front of his arm.
This here.
It's right in front of his arm. I could move it on a second.
Yeah. It's okay, take it. If that one piece is still in it, then we take it. Okay. 1, 2, also only 4, sorry.
As soon as you want it, it comes off the stall.
So,
Almost becomes a barrel.
yeah. I think it's okay. Let me see how late it is. It's okay. I will look at what we have right now because most of the stuff was in the area we wanted. Thank you. If we find a solution.
Last time, before we give up, can you move that frame out of the way? Or wait, does that stay there?
That stays there.
Okay.
You just have to pull away the.
Well, no, I'm saying if I can, well, never mind, because I don't want to land in that area anyway, that's what we're going to do an experiment. I'm trying to think if we can get over there, you can get underneath that rock and do a better job of dirt. We can maybe have more success in propping that up. But I need to go right where that frame is.
Yeah, I would like to go maybe not too far to collect 2 worms that are.
I'll tell you what, I want to
So it's be a little bit. We have like maybe an hour and 15 minutes. For me, it's also important to know what can be done on this place then, no, in terms of collection. So the frame stays and the scoop and the sizzle go in the wherever.
Rail me out, just a little bit. I'll do.
Okay. Okay.
Okay, that's good. That's good. That's good.
Alright, so watch out. Over to John.
That's okay. Drop that that way. It's gonna get back in again.
Alright, so your port jaw is right in that hose for that edge.
I'll see.
I don't know how that would happen.
No idea.
Okay.
Okay, good. So we got that's good. Got the general.
Okay. Okay. We will just go up and around the corner where this big area is, but if we fly a little bit, we will find it. I think it's to the kind of west of the other box. Just going
Behind the box, yeah.
not behind. What am I? We are looking now almost north, no?
Yeah.
Right. And if you just go over these Handtuts and then go west, I think we are at this area where we always collected, and this is the closest because I want the two-worm crabs. Yeah. It might go fast. We look for small two-worms, and they would go then in the firebox free.
All right, we've got a little less, or excuse me, a little more than an hour. So we are going to be looking for a few last collections here.
Nice. Yeah, that's right, because they have a whole canyon, didn't they?
Where we were yesterday, we were thinking 180 degrees out. We went well, yesterday. Yeah, after we were done with the box.
Right, Jeremy, just update your
For anyone that might be joining us for this last little portion of the dive, you are watching live footage from 2.5 kilometers below the surface of the Pacific Ocean at the East Pacific Rise, about 2,000 kilometers to the west of Costa Rica and 1,000 kilometers south of Mexico.
Right, Jeremy.
Because we were sitting like here.
We were sitting right over here.
We were sitting here.
Yeah.
And then we went.
Yesterday we placed an experiment, the first of 3, sort of a form of this little mesh box. You can actually just make it out. Let's see.
So we see
Make it out there. So that little mesh box is sealed to the seafloor, and we're trying to see if anything, any fauna in particular, might be coming up through the venting water that's coming up from below the ground there under that box. But we do still have 2 more boxes to place. So today's dive, we started by looking for a new site for experiment number 2, which we're hoping to place tomorrow. And then after locating that site, we've now been going around taking measurements, readings, and collecting samples.
Like deep, these roots.
As always, want to welcome anyone who is joining us, and we love having folks explore along with us.
Does it have a identifier, the place? Is that waypoint? What's the observation place?
No, but I think I will now when we sit down make the waypoint because I'm sure.
If you happen to be watching and you have any questions, you can type those in the chat, either Facebook or YouTube, wherever you might be watching. And we'll try to answer those as we can.
Yeah, just try to find a good way. I think what we said earlier, look, because I must have or
You want me to log it somehow special?
yeah, but we will first say that's
Put one in if you want.
down and then we make
Huh?
Put one in.
Take one out.
How did, was that 0.9 a second ago?
0.6.
Was that 0.9 when it looked up? Yeah.
I don't know even
Alright, so this is.
I guess in the center of this mound.
I guess we were to the right of this.
There's not really any rules of thumb on samples that cover cruise to cruise. Each cruise kind of has its own objectives and what they're looking for, as well as the permits they might have, depending on where we are and that sort of thing. The thing that's nice about having a remotely operated vehicle is for sampling we can be very precise in some of our collection. We can spend some time looking around for like, oh yeah, that's a good sample there versus that one's all right. Whereas a lot of older school methods of deep ocean collection and sampling, things like dredging, that sort of thing, very non-selective. Obviously you get a lot of stuff, but you might get a lot that's not very usable to you, in which case, you know, with an ROV you can just not collect that and leave it in place. Yeah, we haven't really seen anything eating the worms here.
There's a little square that we were looking at yesterday.
Lots of little crab around, but they mostly seem to be leaving the worms alone. That being said, the worms do have the defensive reaction whenever anything bumps into them of drawing back into their tube. So they do need to protect themselves against something.
I always thought it was because I didn't want to put so many pots. But today it makes no sense to put it in because they're off. If you mix the off with the on, it's like I don't know.
What's that?
Maybe, yeah.
Okay.
This is us, no?
Yeah, that's us, yeah.
And then, okay, this, I don't know how big this is. This is small, but
Oh, there's a marker. There's a marker right there.
No, we're gonna use that one.
Alright, that's a turkey.
Yeah, folks, I've noticed
cannot read it anymore.
the marker there, little bucket lid that's weighted down, and you can see it's been colonized pretty heavily with barnacles there as well.
Can we put on the laser?
Yeah.
So I can see how big they are, because they look the smallest, but I think they're still too big. I mean, they're smaller than those, but 35, or what is this? 30, okay.
Alright, so that's.
Yeah, we don't want to. I think they don't go in the small bio box.
The sampling that was being done next to experiment site 2, where we'll be placing the experiment tomorrow, yeah, we were collecting mostly control samples there, kind of what's in the immediate area. And that did involve digging into the ground there a bit, but I don't think there was a concern of closing up the more porous nature of the rock there. so severely as to block it off.
The only thing we need to do is like
One difference that we do have with experiment site number 2 so far, it looks like, is experiment site number 1, we actually cleared away all the animals and collected them, much like we did that collection today, except we actually did that at experiment site 1, clearing the rock away. And I think they're trying to be a little more delicate with experiment site 2, so that there's not like a crumbled up little pile of rubble right at the site. So instead we were at the next little area over.
I want to call it pie because I don't want to take out what's behind us.
yeah. And this is Alvinella here, and 2 pumps, and up there you don't see it smoking, but maybe it used to be a smoker, and now it should be very hot.
Obviously, it's got the large rifty worms, but is the smaller white, or those Pompeii worms?
This is Alvinella pompeivum, yeah, and you can see a little bit, I don't know, here
Yeah.
coming out. It's basically overgrown, so it's not a black smoker anymore, but it probably was one.
Mm.
There's definitely a smoker behind us.
Yeah.
So I'm going to come up, clear that, and I'll come back down on this area here.
Now, those markers that we were encountering, were those your markers? Were those from prior work here? Or how well studied is this area right now being actively studied?
This study is, this area is well studied. I mean, there were like the last cruise from Woods Hole was, I think, in January, February, March or so.
Updated waypoint, please.
Anything in here you want to look at?
And I think it's also very large. And also, you cannot settle here. No? How? They look very large from.
These are smaller, no?
These are smaller, okay.
These are smaller.
You mean this here?
Yeah.
We go. My idea is just we take a grab from below and we loosen it with the scoop and we grab it.
I was looking over this one to the right, but there's something smaller over there.
But those are gigantic, or maybe this here, but
Okay.
So
Yeah.
I think they're too big. We can look at it. You were there when the tube worms were collected, no? Salva. You were here when they took when so how many did you collect there? Were they all like okay? Yeah, no, now, just now, earlier when I was not here.
The tube of the firebox.
Yeah.
Pilots are currently figuring out maybe where we might be able to collect from, working with the scientists to kind of select the site.
Watch that.
You see something smaller? This is
Just drop that off.
it's only
Okay. Hard to get it from that one.
you cannot.
I can, but
You can,
Oh, this one, yeah.
but
You're set. I want your set.
which one?
William.
Right on below us?
Yeah.
They're pretty-
Small little corner.
yeah, they're
this
long.
here,
No, there's one below us.
You look at
this here.
That'll work? Those- Okay.
I don't know if they work because I have no measure on this. Usually they're way larger than
Lots of eel pouts and some squat lobster in here as well. You saw two of those go across the screen there briefly. So the three primary critters we got here on the view right now, or excuse me, four primary critters. We have those large tube worms, then kind of the brownish golden mussels, the long flexible eel pouts for the fish, and then there was a little white squat lobster that I saw around. There's a couple there on the right.
You need a bigger wire box than your big wire box.
Yeah.
And if I squeeze them in, then
So I have to come, I can't go down where we are. now.
yeah, that doesn't matter.
That
Just
smoke- that smoke goes right there.
yeah,
So
just
it's gonna come around here, go down.
right.
We're safe now. We'll get a close look over here, but we sure don't.
So we good, yeah, so far.
Yeah, no, it was, there's a smoker back there, but
Checks look good, right? So now, see.
what's that? Just turn it off and on. Yeah. See if
I think
There you go.
no, because I mean, you know what it is? I mean, this is like the tube worms are this large in here, and they are covered with kilos of muscles,
Yeah.
and it looks like nothing, but it is like this.
Gotcha.
This is basically cannot.
Down low. Matt, you are now
There maybe in this over there, or
to the right.
no, but yeah, when to follow this, maybe there's somewhere small. Are we? I don't know what is the
Right,
depth.
there's,
2.5. We are really high, you know, because the bottom of the ACT should be 25 or something. We are on, I don't know. We are on something.
Yeah.
We're on something.
We're less than a meter off something underneath us, so I can just kind of follow it back.
Yeah,
down.
right.
Right.
Good.
So keep an eye on. Can you turn my something on? Make some more light, or just open up my iris on my Oh, iris down too. Go up. Here we go. Of course, hit auto on it. Turn to the left, big auto. There you go. I'm seeing nothing down this way.
Yeah.
Alright, something come up behind us. We can't go too much further down.
Okay.
Back. A little rock.
We are here now.
Yeah.
Okay. Then 7. I wanted to go. What were we going? Because the navigation is off a little bit today. So I originally wanted to go to 7, but
7, yeah, so get ahead then.
Well, no, because the map's a little bit off.
we are a little bit
It's
off
not
today.
major.
So I'm,
It's not major. Just go.
There's that one.
huh?
Yeah, there's nothing. I don't know what the offset was.
Yeah, I don't know either.
The only thing we had was experimenting yesterday for
This is
Yeah.
a small patch left here.
Yeah, this one.
There's a big
But
one, there's also 2 small ones
let's-
just above it. Maybe that's
we can go here and just look. But if I cannot fit it in the buyer box, then we just have to search, or we have to come up without anything. So
Oh, we just have to fit it. Firebox.
yeah, that's the problem. I mean, no problem. It's just how it is. So
Not that one. You see beyond, there's 2 little patches up.
I guess we just came from it. We literally just came down.
let's just see this one. Maybe there's something.
Got about an hour left on this one. We probably won't be getting much in the way of mid-water, if anything, on this particular dive, but hopefully we can find some nice samples here before we leave bottom.
Let's see. Maybe there are some small ones in between. We can try maybe these over there. Some.
Okay. Good. On the bottom here.
Well, we have a visitor on the camera.
Okay.
Zoom me out a bit.
This
Try
is-
the well, just I'm on the port arm. I mean, I can try this other one. See what you guys.
I don't know. These seem to be small, but maybe they're just because they're further away.
Always. Okay. Just point again. Which one you are?
The smallest would be the best out here. I think they're smaller, but you need to, to,
Deep.
yeah. Maybe we can pull out a little. Let's see.
No, it's just dead, isn't it?
Ah, yeah, this is dead. Okay. There's one in here. You have to grab it really careful here. There's one in here. This one.
So you just want to grab the tip and
If you, and we can try also to pull on the dead ones, and then maybe some live ones can be hit. But if you squeeze it here, they're dead. I cannot use them.
So the tip,
You have to go
All
to
way
the
at
tip.
Yeah.
yeah, the tip.
The tip.
Yeah, because there's
Yes.
an animal in there. So.
Yeah, I was going right to the base of it.
The base.
Okay, let's.
So go deeper,
Yeah.
to break the
Yeah.
jaw. Get in the jaw.
Yeah.
Monica. Are you taking all the small ones with me?
These are not small, and I have only the small bio box, so I don't know, because we're running out of time and I have no big bio box.
Ah, yes.
So
But we
Yeah.
find no small ones anyway.
This one's really close. I'm trying to.
The
No.
stuck. Okay. Okay.
Can't get a grip on this one. I'll try it out. I'll move the small ones. I'm going to get two. It's really large ones. It's hard to put. It's kind of efficient.
We can also maybe go try on this one here.
Yeah, it's got cleaner jaw.
It's just I believe we could collect it, but if we go below with the to loosen it sort of, no, with the scoop or something, and then grab on something and get a hole. But I don't know if we can fit it in there. We did this leg.
Try this. No. Frame this one. Then we'll go back inside.
Yeah.
of course he doesn't carry.
Go away. See? Yeah, this is good.
Or just,
It's sitting right here now, so
Yeah, you're a couple inches of set.
yeah. Okay.
A little too.
Now, yeah. Just close. There's
Nope.
one
Yeah, he pulled further down.
further down. But
Hey.
just pull. Yeah. Whoa. Wow.
Wow.
There's a little one on it. Wow. Further.
possibly.
Further. See how big this is?
Holy cow.
Whoa. Whoa, this guy's. Yeah, it's definitely.
Then we.
Big fluid.
We will take it still because we will bend it in like this. It's not for life, but I told you they're big.
I mean, do you think
Huh?
so? You think that you could weave it into the small bio box?
Yeah,
Okay.
possibly.
Yeah. We
Okay,
take
so
a
you
few.
launch out and then I'll get to it.
Let's see. I told you they're
That
big.
was the last
This was a
doubt
small
right there.
one.
That was a chuckle and doubt, I think. The chuckle. You might need to move the starboard around with me. It's like
Wow. I'm almost through the thing.
the starboard around. It's clear. I didn't know it was going to catch.
Should be good.
This for sure not. This, you can give it a number and just,
It's sample 30.
yeah. If you log in the sampling, then you have it a long lead. You can make a map. So I don't want to make a waypoint.
Undo the. Don't send on there anymore. Okay.
You did not. You don't even see the end yet of this guy.
No, it's.
Let's take
Yeah,
it.
it's still on. You need to take the bungee off.
Yeah.
Of $5 through.
Okay.
No,
Still on? Oh, right.
I'll get it.
So a few true bombs with a number. And this will be the number Biobox 3, yeah.
Is that John? No, good. So now we're gonna get it back on.
This is the number. No?
30.
Yeah. SO546. 30 giant tube worms folded in Biobox 3, I guess, or something like this.
Where should I put it, do you think?
Well, no.
I can just hold it. Oh, you need both arms
I
on it,
might
right?
need you to use the arm. You don't hang on the units on what? I mean,
What? He needs to open the Biobox with the bungee cord first.
Take it in now.
just need dropping it.
He's not carrying, he's opening the box. Bungee cord. The bungee cord was like there, the box cannot open. It has to be also.
That's the way that he take one to my previous seal.
One of them. We did several things.
Box 3?
All right, so we're going to try and get that tube worm into one of these boxes now. As you saw, it was quite a long specimen.
You can just see it with the hook. See the silver piece? It's in front of it.
You have to share.
Let's do that.
Philip and
I
Teresa.
don't know if it's easier,
But she needs only pieces of the total, so you can dissect it together.
Yes, and all together.
But this is not for Jessie. So did Jessie open hers?
Yeah.
Did she open hers? Do you know?
No.
Okay, then she has nothing to be able to feel.
She told me she would do around 3, but she didn't like
If you go to her, if you find her, tell her I have no collection for her. She might as well keep the others she has in there.
Oh.
You know what I mean?
Yeah.
Because she's not reading probably the, she will get nothing because this is not possible.
Yes.
Alright, I'll go big end first. Obviously.
Yeah.
So I would come out and down. Alright, hold on. Hold on.
Or
Stand by. Whoops.
Can you sit down again? Please? Yeah, can you get the firebox open further?
Nope, that's as far as it goes.
Okay.
Sorry about that.
You can start feeding it towards me.
I'm doing it right now.
Or freeze.
If you want to just grab, alright, so you get the front edge, I'll let go of it and grab the middle.
Okay, I'm just gonna drop this one.
Yep.
It doesn't fall in.
Hopefully not, but me pushing it might help. If you grab it like a foot or so in front of where I have it,
Just gonna do the crab as well.
and take the spot lobster.
That's my fun.
there you go. So when you're happy,
Maybe not too tight
No,
if this is
just touching
possible.
it
Just
all.
touching to
you happy
move
you
it.
got it?
Yeah.
Turn right, yeah.
Alright, I'm gonna let go, okay?
Yeah.
Alright. it in.
Yeah, but you push it out.
Oh, well, you gotta guide it in, dude. I'll feed it to you, you just gotta guide it in. Coming out of the back.
Quite the process getting this tube worm in, as you can see. So our two pilots are controlling both manipulators of Sebastian.
Alright, let's go. Get out.
Yeah, you can try and stop that.
From this side, yeah.
It
This,
just keeps floating away.
Yeah. Just hold it just a little bit right there.
Yes.
A little bit more. A little bit more. That'll be alright.
As long as that's moved.
Sounds good.
there's some smaller ones on it. Maybe we should concentrate on the smaller ones that they're kind of in, and then we just because
Alright, bring it towards me.
yeah. This is good because there's some smaller ones here.
Stop.
We need to deal with that monkey stress now. It's right in our road.
The one that.
What is it? We need everything now. I can hold it all here.
Stand by.
Yeah, it's frozen. Okay, you get good. You can get that over with.
Alright, I'm gonna put this.
Just moving the bungee that's used for securing the other bio box there.
Straight back in.
Alright, I'm gonna put it down right somewhere where it goes.
I don't know. No idea.
Alright. Well, you can work on that while I'm looking at this. I can just hold it. Alright.
got one other piece.
Little guy hanging out and stuff. So.
Well, he's not really a little guy.
Well, relative to what he's had. I don't know if that's is it. Yeah. You come stay underneath and sit up. There you go. Keep going.
So as you can see, it takes a lot of coordination between our two pilots here as they work the two manipulators in unison.
Right there. Sometimes the leaves come up.
Okay, stay safe. He
I
said,
think this
separate.
part of this part of the tube, the ending that is sticking up, is empty.
Yeah, he's gonna close on it.
This also on the side, maybe. Ow!
Alright, it's not close very tight.
Yeah. But what is hanging out on the right side here?
Yeah, that's the very last little bit. Can you grab that?
This is it.
Yeah, but he,
I can open the box back up and you can grab it from inside the box.
I need to find a better side for small tube booms. The other one is also, I think.
Alright, I'll open it up a little bit. Alright, we come up,
okay, what?
just look at the hook.
Wait, hold on a second.
Yeah.
Close it, look at this.
Basically half of the animal is now, I think, outside. I don't know.
This one looks like empty, to be honest.
It looks empty, but I don't know where the animal is really.
Alright, open it up.
I think
Alright.
we will not
going
get.
in. Alright, stand right there.
Okay.
Okay, do you?
Yeah, just as long as
I know,
it's
it's just
not.
Quite a lot to pack into the bio box, but pilot's making pretty quick work of it all over.
Jetzt ist der Fisch auch noch so drinnen. Okay.
It's on the right door.
Hold on, not yet.
Curious eel pout for a moment there.
Push this in.
No,
I can get out of it. Yeah.
Alright, here's right there. Get the,
You deal with the rope.
Yeah.
I'll deal with this. Yeah, I'll let you get the rope. Carry on.
alright.
free close. And then, hold on.
Oh,
Yeah.
No.
no.
There we are.
How to fold I don't know, 1.5 meter long tube in a tiny little firebox.
I think it's like 3 meters long.
Watch the video. I don't know.
That's insane, right?
Are you gonna close the
Yes,
fireboxes
I'm
with the
gonna see it right
bungee
now.
cord also?
Yeah,
Yeah,
do
so,
it right now.
yeah.
Okay.
Yeah.
I'll do this one.
This one is. Highlight video. I did a highlight video.
The Monidops is still in there.
No,
Yeah.
the guy got out.
Oh, did he? The little squatty?
I think he's still, is he in there?
If he's in there, then we have him.
Yeah. Yeah, I see him in there on the sit cam. He's in there.
That was
Alright, yeah, look at your size there. Just put it there. Keep going up. Keep going up. Alright, bring it in. Yeah, a little higher. A little
No.
higher. Nope. A little more, a little higher. Alright, there you go. You're good.
What time is it now? Okay.
Okay, so that's how it's sealed and that's how it's sealed.
Ham.
Wait a
We have
So
half an
no.
hour.
Oh, we got another one, sure.
No. We open the box and get another one. No. Okay, thanks a lot.
Suction samples available.
So it might be a little bit difficult. A little rock in a coral quiver.
Alright, so
If we have
start
time.
around, yeah.
Yeah. Okay, no
Well,
rocks.
a rock from here or somewhere else?
We can take it from here. If you can grab one and see one, then it's fast.
Take a male. We need males.
A male
We
rock?
are the-
Not male.
No, I cannot. If this large one is not a female, then you're out of luck. We cannot possibly fold the second one, and this is not possible. And
Yeah, so she wants
I
to put it
cannot-
in.
yeah,
So a good idea.
Would it be, do you want it for the rock or do you want it for animals that are on said rock?
just a rock.
Okay,
This is just
you might do it. Yeah.
can be something if you
See what I'm looking at.
whatever. You,
There's a rock right there below, yeah. So you pick that one up?
It's got bag for the color.
I think we got a milk crate right there.
You can't go in there.
now it has to go in the coral
Oh, okay.
quiver for the bacteria.
Gotcha.
Let's see.
That's pretty big.
A tiny little piece is good also. This one, yeah. It can be really small. This is good. If you,
Super fragile.
there's another, yeah. Wow. Here, the one you lost is this here? You have to close.
Loads of beds. There you
You
go.
could take wrong.
Tons of beds. It just becomes smaller beds now.
It's a really cute fish.
This rock is not a great one. Can we come back a bit, Chris? Just
What do you mean back? Like
well,
5
like
X?
some space. 'Cause there's
I mean,
everything here is just like.
I'm not really sure what I'm sitting on, so
Alright, fair
we'll
enough.
figure. No, I'll back up. I just don't
I'm
know.
gonna look at this one and see. One of them heavy. Just don't look sure.
I can come back. There's that one just underneath the rail.
Yeah.
I don't know if you can reach it.
Not from there. Unless you pull the rail back.
Yeah, that's not gig. I ain't doing anything.
This here?
No. Hold on.
Ah, okay.
Okay, it is holding me in. Hold on. Hold on, I have an idea. Alright.
Because I picked the biggest one.
Yeah.
Break.
There you go.
That's just powder, man.
Alright, I can.
I'm gonna try to use the rubber
Yep, I
gloves.
can fly off and come back. So.
The rubber
Rubber, yeah, we're trying rubber fingers.
is
and see.
better,
Yeah, it should be.
I guess.
Could you use the scoop to collect this?
Yes,
rock?
we can, but we don't have to. We have to before we cover him.
Let me go to the left here where there's a bunch of little rocks.
It's possible, yeah, I can just use the
Is it okay?
Sure. Any little rock. That's a lot of rock.
Oh, could have gotten that little one, huh?
It's okay. No, now it's with a full. I don't know. You cannot be so picky in the end of the dive.
Yeah, one down.
You cannot be so picky at the end of the dive because
What's that?
I said you cannot be so picky at the end of the dive. I cannot. I could not. You know.
What? No, he wasn't inside there.
Yeah.
Not possible.
That was crazy timing now.
I was like, what? Doesn't make sense. So I had to do that.
So
Alright, the world is your rocky oyster.
Right, just leave it as is. I go with it.
No, positive one.
The one right above you, next to that dead worm
yeah,
underneath.
just wait till you
Just
roll
below you,
it.
6 inches to the right.
If you grab one.
That's what you just do this thing.
There we go.
There we go.
Got up to 5.
Yes. Well, 5 on the side.
I know, I see it.
Oopsie. Let's. We still have time.
Yeah.
No problem.
Just leave it set up.
Yeah, yeah, yeah.
That's a big one that was it breaks. The quivers are about the same size as the jaw opening, so. Well, this one right here has.
It's right in the right.
I'm ready in.
Mm-hmm.
Better than I thought it was. Shadow is just above it now.
Peek it a little bit in. She wants
There
it
you go.
crumbled anyway.
Second?
She wants it
Nice.
crumbled anyway, every, so this is
She
perfect.
told me.
Yeah, but I cannot ask you to collect it
Would you like
differently.
another piece? Well,
Thank
yeah.
you.
I may as well get another little piece.
No, this is enough.
Sure.
She doesn't need much. No, this is enough.
20 minutes, what do you want to do?
We're gonna fly around a little bit before we have. So, if I see like this area where we came from, the outskirts are with these barnacles. Yeah, there's something we can, if we see something, we suck up something, or we, if possible.
Okay.
Otherwise, we just look. Because we have everything basically almost secured now, so one more thing, if we find something.
Okay, there's I still got chisel in there. Do you want to throw in the box?
Yeah, we can grab it.
This is like,
Portion out for you. Don't get any ideas when you look at the box.
tiny this. Top it.
Here to help.
He was a coffee. Okay, those two things we have. Alright, okay. This is this was the sample 31, no?
Yes.
Okay. Okay. You can just write the observation raw collected where large tube-worm or giant tube-worm was collected. So basically this sample 30, and they can look it up, the long lead then. Ah, there's an octopus. Look,
Oh yeah.
volcano octopus.
Alright, so what's the sugar on that one?
Okay.
Can we just watch it for a few seconds?
Yeah, we'll just
Yeah.
trying to zoom in hard on at least the part.
Right now?
Yep. Alright. Monica, those need a drink now.
Oh, it's fine. Wait a second.
What?
Never mind. It was swimming and then it decided to get this swimming.
Ah, okay.
Alright.
Okay.
Alright.
Actually, this here. Let's go here. Yeah, because it's very close and I'm curious if something is on it.
Okay, I don't know how long we go there. You'd have to get over there too.
Awesome.
Once that land down somewhere.
That's not, you gotta find that chip or
Are you using this camera? Okay, then I just see. These dog's barnacles are cool.
Eight particles in Spain, eight particles in Spain, they were really good. It's barnacles for dinner a couple times in Spain.
Where?
In Vigo, Spain.
Oh, okay.
They were really, really good.
In the south, they don't need it.
What's that? Barnacles?
Mm-hmm.
They're right.
I enjoy them.
That's it. Either or.
Okay.
Felt like milk.
Well, they're not in the
I know, but.
What are 7 dollars?
No, I'm looking for some snails, but
I get a little closer.
it's okay, I would see it.
Good eye on the octopus, by the way.
Hmm?
Good eye on the octopus. Good spot.
Hmm. You didn't see it?
Not that you said something about it.
The good thing at this hydrothermal vents is that the species richness or the number of species is relatively low. So compared to like a coral reef or so. So it's not so difficult to know all the species after a while. Okay. You can I don't see any. I was looking for something special, but I couldn't see it. Goes like here. Okay.
Yeah, I take it that wasn't on purpose.
No, that was yours.
There's another bunch of barnacles here. Let's just check it out. Here we go. Jan,
Yeah.
look. This is not the one you want.
No, it's but maybe.
Yeah.
The snake?
This is so much stuff. Doesn't want to, it focuses on the, okay.
What is this? What is this?
Okay, let's see.
This.
I go in there, yeah. This is the, shit. Okay.
I got it.
No problem.
Is the siphon of
Yeah,
mushroom?
but of a very large one, it only one. Click here, this here, basically it's the same as this here. But this is this muscle, and this is already, I mean, I don't know, 10 centimeters or whatever, and this goes behind. Okay. If you look close, you really see that overseas here, oh, everywhere. But Daphne I have not seen. Oh no, this is our, schau mal. I want to get him right. Don't disturb him, I want to
say.
take it.
Let us do the camera.
I've never seen this fish eating.
No, I've seen it.
You?
Yes.
You saw it? I've never seen it. For me, they only sit around.
They are very abundant. I've never seen
I
them
know.
so abundant. Like
But yeah,
here and there, one, you know.
no. I think they are really abundant, but I've never seen one eating. What do they feed on? You saw the mouth movement of eating, but you didn't see what? Yeah. I think they pretty much eat everything. I mean, scavengers and predators, but really.
This was that grumpy old man.
Takes right away.
I mean, okay. Last minutes for bye bye. Let's see if we find something else. Okay. We have I think I looked at those already. I cannot find this gastropod here. Huh? The ones Jan was looking for, the white ones that are quite big really. I don't know the species. but I know how they look, and they were on the circuits.
Oh, for the piece? New Year, we fortune in, railroad. I don't know if that. That's the only annoying one like that. Yeah. It's not bad.
Okay. Those were also. No, but I just wanted to have a nice view on this. So earlier, like they were super very hard to found, and
It's the first time I see like
so many.
and barnacles. Yeah, also
Yeah.
like I've not.
I think I saw a few, but very far outside. This is also a species that moves in very late in succession. But then once they're there, they're there.
Because the temperatures that you measured at the tube rooms, they're also lower than what we had in the other years. So in 2006, 7 it was like much higher. Also like the collections, you know, the first ones I ever looked at, they had also like more than 20 degrees.
Mm-hmm. I just love this camera where you can zoom in and you have the feeling you're sitting under a microscope. This is so great. Okay. This was not so good. Okay. I think we can leave bottom.
Call it?
Yeah, yeah. Quite a successful dive.
Bridge RV, so you know that's about to come off bottom and it's not commenced recovery.
Alright, ready? Coming out bottom now. Okay.
Thanks a lot
Alright.
for the hard, very difficult collecting rocks and other stuff. Difficult. Thank you.
Okay, thank you guys for watching our stream. We will be back tomorrow with another dive. See you then.